All right. So science, we're getting into it now. Welcome, everyone. We're excited to start this first session. I'm going to be kind of brief to try to get us a little bit back on schedule, but we're going to be talking about marine biodiversity and ecosystem resilience. This session was organized by myself, Susanna, Victoria, Dreux, and Tricia, and we've been working on it over the past few months, and we have so many exciting speakers and lightning talks and first ever debate, as Dreux mentioned as well. So make sure you have your coffee and your tea and you're ready for a lot of interaction. And the goal of this session was to start talking about kind of observational evidence that we have in the ocean about understanding all different types of diversity in an ecosystem and how that might relate to resilience in some way. And each of our speakers are going to provide kind of a large breadth of knowledge in their own way of defining both diversity and also resilience in their own way, which they'll talk to us about. And we hope that in our debate, we can start talking about, what is known and unknown about the link between these things. And we hope all of you will be able to contribute to that. And again, we're just kind of hoping that we can talk a little bit about how diversity can span many different types of scales, which I think our speakers are nicely covering. We're going to talk a lot about what does this diversity actually mean? Is it taxonomic, functional, et cetera? And how do we need to change some of our tools perhaps to better address that in the future? This is also, I think, really interesting and ties nicely into some of our other sessions that we're going to have throughout the week. And in fact, links even to this new area that OCB has been focusing on lately related to marine carbon dioxide removal. Where folks have been talking about not only carbon credits related to carbon dioxide removal, but also even biodiversity credits. So what does that actually mean? And I think, as Heather really nicely mentioned, many of the kind of future programming that OCB does is born from this summer workshop. So we also hope that this session can start to lead to lots of discussions on this topic and maybe even launch some OCB activities in the future. So again, the goal of this session is to start talking about the state of the knowledge and all understand the terminology and definitions within this field, as well as thinking about the gaps and how they can be filled and how we can move forward, and if we need to have some follow-up activities after the session as well.
All right, so we're going to be having a debate the first time at OCB, and the goal of the debate overall is to challenge our preconceived notions about the roles of biodiversity in the ocean and what it means for resilience. So there are a number of different concepts that we thought of that we might want to talk about in the debate, but you shouldn't be limited to thinking about these different topics. But how is biodiversity changing in the ocean? How are those changes likely to affect resilience and other ecosystem processes? Have we measured a link? Like, have we quantified the link between diversity and resilience? If not, how could we? How would we? How do we define scale with respect to resilience? What level of community or individuality is relevant to the role of diversity? What levels of diversity should we focus on so that we can actually quantify this impact on resilience? Are higher level attributes of ecosystems predictable or not? As Randie just pointed out, there's a movement that you can now get credit for assessing your impact on diversity similar to climate credits and other things. So we have to think about how these guidelines are set to be applicable, and really, as ocean scientists, we need to be involved in that conversation. And so we need to understand how we think those credits should be applied or if they should be applied. And, other topics, what is resilience? What makes microbes resilient? Can ecosystems themselves be resilient? So the debate process. You have homework as you're sitting here absorbing these wonderful talks. You want to think about the things that I just listed as concepts to consider, or what other concepts and topics you think need to be discussed, again, targeting this goal of challenging our preconceived notions about biodiversity and the ocean and what it means for resilience. We'd like you to write down your thoughts. There are index cards that are being passed out. There are index cards that are at the back of the room. You don't have to fill out an index card if you don't want to, but if you do, we want to make sure that you get your thoughts heard. So you can write down your thoughts, opinions on the index cards. It'd be helpful if you want to share your name with them, because then we'll have you actually be part of the debate. But if you want to submit an anonymous comment, that's fine. We can read them for you. Pass your index cards to a session organizer or place them in one of the boxes at the back of the auditorium. So there are 2 boxes. They're a little like milk jug type on one side and a box on the other. Very, very official. We like to recycle. And, you know, so our session runs for a while. We've got a break, we've got lunch. Try and submit those index cards at least 20 minutes before the session restarts after lunch, just so that we have a chance to look at them, so that we can group, the session organizers can group the comments by the theme. Our MCs of the debate will call people up to read their statements or read anonymous comments, trying to group them together by topics, and allow people to either present some of the opposing or corroborating comments, if we have some, or solicit the audience to participate as well. So if you don't have a thought to put in an index card until you hear someone else's read out, you're not done. Like, you can still raise your hand and be part of the debate. And then we'll try and moderate it to make sure that, you know, not one person is talking the whole time, to make sure that students are getting heard, et cetera, and move on to topics as we feel we've sort of finished the debate. Or if one topic is taking up too much time, we'll tell you to talk about it later over at the poster session at some point in a corner, or, you know, at the next lunch in a dark corner. But we want to try this out and see if we can start talking about this in a broader sense with the community. All right. So I'm going to let this.
Hi, everyone. I'm Tricia Thibodeau, and I'm just going to set us up briefly for our talks today, just to give you a sense of where we're going. We wanted to keep things fresh, so we're going to be doing a mix of plenary talks as well as lightning talks, which are 4 minutes each throughout the morning and afternoon, and then we'll have our debate. And so just to give you a brief overview, our first talk will be with Emmett Duffy, and he'll be really setting the stage for us, thinking about biodiversity and resilience. And then we'll have Tammi Richardson from the University of South Carolina, focusing on phytoplankton. And then we'll have a series of lightning talks. And then we'll transition back to our plenary speakers with Gwen Hennon. And then we will do some more lightning talks focused on a range of different topics, from microbes to thinking about how things are getting smaller in a warmer ocean. And then we'll have a few more lightning talks just to keep things interesting. And then we'll wrap up with Jessica Labonte, focusing on disturbance and how that influences biodiversity and resilience. And then we will have our wonderful debate. So in an interest to keep us on time, I'll wrap it up there, and I think we'll go ahead and start with Emmett.
Good morning, everyone. I hope everyone can hear. I usually don't have a problem speaking loudly. It's a great honor to be here. I was one of the people who had to stand up as the first time I'm here. Pretty sure I was the oldest one in that group, so sorry I'm late. But yeah, it's truly an honor to be here, and especially a little bit intimidating, I have to say, to be the first science speaker in this, because it's the first time, and also because of this debate thing, which I thank you, Dreux, for the explanation, because I'm still not sure entirely where that'll go, but I look forward to it. So my job is to introduce this session on biodiversity and resilience. And I want to really frame this around the question of what, if anything, does biodiversity have to do with ocean carbon and biogeochemistry? That's a straw person, obviously, because many of you are really on the forefront, actually, of understanding how biodiversity influences carbon and biogeochemistry, whether we call it biodiversity or not. So kudos to you, and my others in this session will be really getting down into some cool examples of that. So to do that, I'll attempt to set the stage, as I say. This is not an academic question, obviously. The intertwined crises of climate change and biodiversity change are really sort of the center of what we're dealing with now in the Anthropocene, as has been mentioned earlier. Just to give you a preview, I will answer this question with a definitive yes. It's not really a yes or no question, but I will say there is resounding evidence for how biodiversity influences ocean carbon biogeochemistry resilience. And I hope to give you a sense of that as we go on. So I want to start at the largest possible scale. Everyone knows this figure. It illustrates beautifully how the shape of the planet and physical processes drive biology and biogeochemistry. Visible here as the surface chlorophyll, which is, of course, the origin of all organic carbon, at least in the ocean. Not all, there's some macroalgae as well. And thus of biogeochemical processes. Productivity will figure largely in what I talk about today about biodiversity. So we now have a pretty clear and detailed picture of this relationship on a planetary scale. Specifically, again, the distribution of continents and oceans, the abiotic environment really shapes the distribution and abundance of organisms throughout the world. And that feeds into biogeochemical processes. But of course, those relationships are also bidirectional, even with geomorphology in the long run. That is that biology feeds back to influence the environment and mediates the ocean's biogeochemical processes. So a central question is how important is biodiversity relative to other factors, climate warming, other kinds of disturbances, and how do they work together? That's what we'd like to talk about today. One way to approach this, again, from a very large scale, is to consider the Earth without organisms. Lovelock, famously back in the day, sketched out what the Earth might look like in the absence of life, and it's more or less of a nightmare. Blazing hot on the order of 300 degrees C, intense pressure, anoxic, and so on. So what I think that points out is that on a planetary scale, at the largest scale, life is a driver as well as a passenger in biogeochemical processes. Yeah, that's what we'll talk about on a slightly smaller scale as we go on. So that planetary view gives us a very interesting perspective, I think, but it also obscures something very important, which is that life is not a thin green film on the surface of the ocean and the continents. At some scale, obviously it is, but it's a spectacularly rich collection of very distinct life forms, millions of them, that are distinct in form and function and are doing different things and interacting with one another. So what does this mean for how the ocean system works? That is our charge. And specifically, how does that influence resilience, by which we mean the ability of the system to deal with challenges, disturbances, and bounce back, very, very generally speaking, including the profound changes that are underway right now with changing climate and environment, as we've mentioned a couple of earlier speakers. So I think the best way to start this is from a conceptual or theoretical standpoint. Why should we expect diverse communities to function differently? And I'll talk specifically to begin with about productivity. Why should they be more productive? And the first part of that answer is that all species are different in form, growth rates, environmental tolerances, and so on. And that's not coincidental. That is a inevitable outcome of the competition and interactions that organisms have as they are developing over the course and evolving over the course of millions of years. So that functional differentiation among organisms is fundamental. It's also true that the environment is highly differentiated on all kinds of scales. And if we look at that in the lower left here, I can use the old school pointer. If we think about an environment in only 2 dimensions, and I'm using prairie plants here because this is what most of the research in this field has focused on in the beginning. If we think about water availability and light availability, a 2-dimensional sort of environment, those circles represent niches of different organisms in that 2-dimensional space. And you can sort of see how that might work with the prairie plants due to the depth of their rooting, the way that they spread out their photosynthetic surfaces above ground. So, thank you very much. Yeah, thank you, Victoria. So the environment is variable over time. When we combine this differentiation of species with the environment over time, the species begin to sort out into the different niches. And so that there are 2 ways that diversity of a plant assemblage here can contribute to higher total community productivity. One is through complementarity. That is, the species are occupying different parts of that niche space and therefore capturing resources more effectively together than they would alone. Complementarity. The other way in which more diverse species can be more productive is through a simple statistical phenomenon. Sorry, don't know what got into me. of so-called sampling. That is that a diverse community is more likely, simply but for statistical reasons, to contain the one species that does better and can take over under those circumstances. And we'll see an example of that in a moment. So these are a couple of the mechanisms by which this can happen. So early experiments confirmed these general predictions in a variety of disparate systems. I'm showing 2 here. Again, much of the early work was in grasslands or prairies. You can see that these are from experiments where plants where fields were planted with somewhere between 1 and 32 species or so on. And you can see there's clearly an increase in average total plant productivity on the plot as diversity goes up. Similarly, another example of very disparate in an aquatic system, eelgrass systems with small grazing invertebrates. Grazer biomass also is higher in treatments with more species. So similar patterns, different systems. Over the last 20 years or so, lots of ecologists have done lots of experiments like this, and the results are surprisingly consistent. On average, more species lead to higher total community productivity. That's in aquatic systems, in terrestrial systems, different trophic levels, and so on. There's actually not much difference in the shape of these curves between aquatic and terrestrial systems, despite very different kinds of primary producers, for example. However, we do tend to see a little bit steeper relationship at higher trophic levels, that is for herbivore biomass production as opposed to plant biomass production. So how does it work? What is the basis of those relationships? Coming back to the seagrass grazer example again, we can see that in this case, the yellow triangles here are the grazer biomass production for single species treatments. Each of these different little bugs, amphipods, isopods, snails. And so you can see what's happening here is that there is a sampling effect. That is, within these small mesocosm tanks, there is one species that does best. This guy up on the top. And so this is an example of a case where where there has been selection for the one species that can take over. That is much more likely in these small experimental systems than it is out in the wide world, as we will see shortly. So this is sort of reminiscent of how agriculture works in our activities. We find that one species that produces best under the circumstances that we like, and we grow it in gigantic monocultures. So in experiments, biodiversity, on average, is pretty consistent in increasing productivity. But obviously, experiments are not the real world, which is bigger, more complicated, more disturbed, and allows these ecological processes to run over much larger timescales. So the question is, does biodiversity have a similar effect on productivity in the real world? Answering this question is obviously harder than in experiments because we do not have experimental controls. So what I'd like to do is just explore one example using marine reef fishes. So understanding the productivity of fishes for food was really the birth of marine ecology in the early 20th century. And what we're using here is an amazing database that has come from the Reef Life Survey program that's based out of University of Tasmania. Graham Edgar and Rick Stewart-Smith run it. This group and colleagues have conducted something like 5,000 surveys using standardized protocols of reef fishes on both coral reefs and rocky reefs from, Greenland to Tierra del Fuego and all of the coral reefs in between. And so because these are done in a standardized way, we can ask what can we gain, what can we get out of this database to understand how fish diversity is influencing total fish biomass. So first thing we do is look at a simple bivariate correlation, as shown here on the right, and we can see that there is indeed a strong positive relationship between the number of fish species in the system and their biomass. Now note that both of those are on log scales, so there's a very large variation here. But of course, there are lots of other factors that are influencing fish biomass between the Arctic and the tropics as well. And so without that experiment, we need some kind of statistical control over the other drivers that are happening here, of which there are many. So to do that, we used structural equation models, which basically allow us to link these various individual models together in a path network of potential influences, and we can use that to structure that path network the way we think is happening, and then test the different components of it. So the result, to make a somewhat long story short, is that fish diversity strongly affects total biomass here. We can see even when controlling for mean annual temperature, range, chlorophyll, various nutrients, depth, salinity, and so on, we have strong impacts of fish richness or diversity and functional diversity on fish biomass. Now, not surprisingly, temperature is still a strong indicator, or sorry, a strong driver, but it's perhaps a bit easier to compare these by summing all of those paths and showing them over here. And what we see is that the standardized effect size, the relative influence of biodiversity is comparable to that of temperature across this large global range, which I found quite surprising personally, but that turns out to be not an uncommon finding, as we'll see. I won't go through all of this in detail. I just put this up to show that result, finding that in the wild, in open observational data sets, there is a strong link between diversity and productivity, has been found now in a wide range of systems. On the top, we have terrestrial, on the bottom, marine. So the grasslands or prairies we saw earlier, dry lands around the world, global forests. And down below, we have estuarine phytoplankton on the left, the fishes that I just showed you, and even deep sea benthos. So there's a strong consistency in what we're seeing, and all of these were analyses of observational data that controlled for various other environmental drivers. So what's going on? How does this work? We can get some sense of the mechanisms of this from some really interesting studies on phytoplankton. So this is from work by Strebel et al., who showed that in the far left, in both experiments, which are blue, and in observational data sets of the field, which are green, the more diverse phytoplankton assemblages also had a broader variety of pigments, which are obviously the light capturing machinery of the cell. And in the middle, as that pigment richness increased or was higher in the higher diversity assemblages, we also see that there is more efficient light absorbance. So that in the end, the more diverse phytoplankton assemblages had higher specific primary production, which is probably a similar thing that's happening in terrestrial plants as well. So all good so far. Diversity, on average, leads to higher productivity. Fixing more organic carbon, which enters all of the biogeochemical processes we're interested in. But what about resilience? What we're really focusing on today. So something like 40 years ago, Paul and Ann Ehrlich introduced this famous analogy of comparing an ecosystem to an airplane and the species to the rivets that hold it together. And their analogy was that as you pull rivets out of the plane, which is kind of what we're doing with species in the world right now, the plane's okay for a while. You can lose a few of them, but at some point you cross a tipping point, the wing falls off, the plane crashes, and that's not good. So similarly, obviously, so what we're hoping is that that's not what's happening in current ecosystems, but we are in fact taking species out, or at least reducing many of them to the point where they're not able to perform their functions anymore. So, a nice thought experiment, but is it true? Well, let's look at some data again. But first, we need to deal with the question of what we mean by resilience, which you may have noticed is used in very different ways by very different people, even by the same people. It's more complex than productivity. And what I'll do here is just introduce a few components to what is often called resilience that may help us in looking at the data. So the first is simply the stability through time of a system, the inverse of the coefficient of variation. You know, during the sort of normal slings and arrows of life in the world, how much does whatever your estimate or your performance metric may be, whether that's productivity, total biomass, whatever, how much does that change? We call that stability, or strictly speaking, it's the inverse of stability, right? How much it works. The second important question is what happens when there is a disturbance, a storm, a heat wave, whatever it may be, an ocean acidification event of some sort. Oftentimes, that will decrease the system's performance, and the distance to which it, the magnitude of that change, let's say, is the inverse of its resistance. So if the system changes a lot when the storm comes, that's high resistance. If it doesn't change much, low resistance. A third, I should say, aspect that is sometimes strictly called sort of the narrow sense of resilience is how long it takes for the system to bounce back to where it was before. And that's what we'll call resilience here, the inverse of return time. So we can start to explore empirically how this works by returning to grasslands, again, where most of this work is done. Forrest Isbell and colleagues synthesized 46 biodiversity experiments that have been done in grasslands, North America and Europe, and that have run for a long enough time that we're able to look at their responses to big changes in weather. So extremely hot years, dry years, floods, and so on. So the question is diverse vegetation more resilient, using those different components, in the face of climate shocks, something obviously we're very concerned about these days. And the short answer is that diverse vegetation tends to be more stable through time and also more resistant to climate checks, meaning that it doesn't change as much when those shocks happen. Not necessarily more resilient. In other words, they did not see evidence that it comes back faster. So what you're seeing here, the small individual gray lines are the trends in individual experiments, and then the means are shown there with the confidence intervals. More diverse vegetation, more stable, more resistant, not necessarily more resilient. The red and blue are for dry events versus wet events. So there is some evidence of resilience to drought in these situations. Those are all experimental. The figure on the right looks at the same question in observational data across terrestrial plant communities all around the world. And you can see that ecosystem stability in this case, similarly, is higher in vegetation that is more diverse. What about marine systems? So let's go back to the global fish survey data that I talked about a moment ago. We do not have a time series for these data. So basically what we did was to divide those survey data into sites that had high richness versus low richness and of fishes, and then compared how fish biomass looked across a gradient of annual temperature variation. So in sites that were highly variable versus sites that were relatively stable. And what we see is that the fish communities from low richness sites, that is few fish species on the left, had a much wider window of variation in response to annual range in temperature. That's the red bar here, compared with the high richness sites. So some evidence that fish diversity confers some ability to is more stable in the face of changing weather or climate over time. Just a few more examples of resilience in the marine realm, which I won't go into a lot of detail in, but there have been a number of experiments and modeling studies that have addressed this issue of how diversity influences resilience, various components in marine systems. So the first on the left there, classic experiment by Jay Stackowitz and colleagues, where they created sessile invertebrate fouling communities with different diversity, put them out in the field to look at their resistance to invasion by new species, including non-native species, and found that as the richness of species in those treatments increased, there was less success of invaders in getting in. And this is largely because the more diverse communities used up more space. Again, going back to our grassland model with the two-dimensional habitat, what more of that niche space, if we might call it that, was taken up in the more diverse systems. So more resistant to invasion. Secondly, and this is the only example I'll show that uses genetic richness as opposed to species richness, this is work by Randall Hughes and colleagues, which is where she planted eelgrass with different genetic richness plots to watch how it changes and grows. And this is my personal favorite example of a graduate student snatching victory from the jaws of defeat after a bunch of geese came through just a couple of weeks after she planted them and ate all the grass. And of course, she went through a dark night of the soul, but decided, well, let's get out there and see what happens. And sure enough, when the grass started to grow back a little bit, she found that survival was highest in those plots that had the highest genetic richness, which you can see here. And that was true early on and remained true later on. And this group, Randall Hughes, Jay Stackowitz, have done beautiful work in this area on seagrass genetic diversity. So the final, the sub-final example that I'll explain here is from fisheries in the Amazon basin, so not marine, but aquatic fisheries, where there is actually a really long time series on fish yield over time in different sites that vary a lot in fish richness. And basically, using that time series data parameterized with the empirical data, these authors did a model of how that system responds and found that the fishery is much less likely to collapse under at high diversity. In fact, at the highest diversity, they couldn't get it to collapse at all. So this has an influence on people, and I'll just mention one other example of that, that fishers targeting diverse species similarly in Alaska were more resilient to the major regime change that happened up there in 1989, changed the oceanography, also changed the market conditions, and you can see that those communities that captured multiple different kinds of organisms, a high diversity, sort of broke even, whereas everybody else had a very bad year and continued to for a while. So moving right along, we'll just go to some questions that I've been thinking about that I would argue are sort of at the frontier here. You know, I've talked a lot about grasslands and some other things. I know many people here are rightly thinking about the open ocean. How should that differ than what we're doing here? Well, the ocean is three-dimensional as opposed to the two dimensions that we see in terrestrial vegetation. The ocean, that means that competition is weaker. We can talk about that if necessary. The ocean is big, obviously. Currents are moving things over large distances. We have vertical transport out of the surface layer that can have a big influence on ocean carbon and chemistry. The other thing about the ocean being big is that much of it is also invisible, which is not the case on land. And as many of you know better than I do, there are big challenges to seeing important processes in the ocean through remote sensing, which is done routinely on land. Ocean life is very small. Instead of oak trees, we have tiny microbial producers, and that has big effect on how fast things can happen. So many of these can factor in. So I will stop here just by mentioning that our marine geo program at the Smithsonian that I lead is really focused on trying to address a lot of these questions by bringing people together. We've heard a lot about networks here, and I loved Heather's talk. I'm taking notes about how we can do what we're doing better. So these are the kinds of questions we address, and I'll also just mention that we are helping to organize the Marine Life 2030 program through the Ocean Decade. We will be at the World Conference on Marine Biodiversity in Malaysia in July, and would love to talk with anyone about how to get involved in either of these. So thank you very much, and I look forward to debating.
We have maybe time for one quick question. Do we have an early career scientist who has a burning question to start to kick us off here? If not, it'll be a free-for-all, so this is like your your countdown time. An excellent early career scientist.
I didn't hear you. I was thinking. No, I was thinking about the issue of mostly being prokaryotes in a marine environment, and the concept of species that you have been talking right here. When you go to prokaryotes, changes dramatically. So how you address that?
Yeah, so species, much of this work is focused on species because people know how to identify them. But what's really important is trait diversity, right? And species are, to my mind, sort of a proxy for trait diversity because they're convenient bundles of traits. But for many species, there's a lot of variation within the species, as we saw with the eelgrass. And for prokaryotes, that's especially true. And so whether you call them strains or species or something else, to my mind, the important question is how is what they do differently influencing that aggregate performance?
All right, well, I think with that, we'll go on with Tammi Richardson.
Good morning. All right, thank you so much to the organizers for inviting me here. I was tasked with talking about biodiversity from the phytoplankton perspective, or the invisible perspective, perhaps, in the words of Emmett. What a huge task. right? So please bear in mind as we go through this, I picked and choose as examples as I see fit, but really what I'd like to talk about in general today are our friends, the phytoplankton. I tend to move around, so is it okay if I move away from this thing? Okay, good. Can you hear me okay? Oh, good, there we go. So you've probably seen phytoplankton before. They are incredibly diverse in terms of both structure and function. They can be anywhere from 1 to hundreds of microns. They can be all sorts of different shapes and sizes, spheres, spines, chains, big globs, for want of a better description. Autotrophs, mixotrophs, nitrogen fixers, calcifiers. We'll talk about all of these terms and how they relate to biodiversity in a second. And all of these vary greatly in their elemental requirements, how they respond to light, how they respond to temperature, okay? So I think I'm going to start by talking just really briefly about why as oceanographers should we care about phytoplankton diversity. Really, when it gets down to it, knowledge of biogeochemical cycles and the structural and functional diversity, knowing that is key to understanding biogeochemical cycles in the ocean, including organic matter export. And I think Emmett just mentioned a little bit about that. And boy, Emmett set me up really well for this because I have one statement that maintaining biodiversity is also incredibly important for marine ecosystem stability. Particularly in changing environments, and I'm going to focus on those. And it may even increase productivity via a functional redundancy in space or time. And by that, what I mean is when one phytoplankton type, I'll say for now, is maybe not performing well, another one may be doing better in terms of photosynthesis, growth, or any other, insert your favorite physiological function here. We assess phytoplankton diversity on many different scales. If you are work with what I call the real microbes, the really tiny, itty-bitty green things that are hard to distinguish morphologically from something else, you may be more interested in the genetic or the phylogenetic diversity. There the question arises is who is different from whom, and how do you establish those boundaries for what is a strain versus a species, et cetera. Species diversity is a little bit easier for me to describe in terms of this is the morphology. We do this via microscopy or via what I'm going to call the flow methods, flow chem, flow cytobot imaging, flow cytobot, all of the different imaging approaches. You're going to see this later, this term Shannon-Weaver index. We very often calculate some index of diversity, species diversity, using data that we've acquired in this way. We may also talk about algal group diversity, diatoms versus dinoflagellates, chlorophytes, cyanobacteria. We've maybe distinguished those based on HPLC-based pigments or spectrofluorescence or some other technique. And then there's functional diversity. There's two, in a one-week comb of the literature, I could find really two different types of functional diversity in the literature. One is a cell size-based estimate of functional diversity with the idea that physiological functions often scale with size of the organism, size of the cell. That's often quantified by Rao's Q, that's the index here that's analogous to the Shannon-Weaver. You will find in the literature discussion of phytoplankton functional types, or PTFs, things like organisms that function differently, they may calcify, they may fix nitrogen. So this brings me to what I guess I'm calling challenge number one of actually characterizing and quantifying phytoplankton biodiversity, is that characterizing and counting everything in the water is hard. It's still hard. We've developed all sorts of different techniques, but to this day, we still don't have the one magic probe or magic dipstick that we can stick in the water and do everything from the picocyanobacteria all the way up to the microphytoplankton. Instruments vary with respect to their basis of measurements. So we've got optical instruments, electrical-based, image-based instruments, and they differ slightly in how well they perform in measuring cell size, cell abundance, or taxonomic characterization. I pulled this one from one of Susanne's papers because I love this one where she did an instrument comparison, the flow chem microscope, a BD flow cytometer, and two Beckman-Coulter multisizers. Generally speaking, they did this with 3 different species, cultured phytoplankton species of various sizes. And generally speaking, all the instruments did pretty well in terms of agreeing with the abundance and the cell diameter estimations. But they did find that the flow cam and the microscope, the ones where you can literally look at the organism, were a little bit better at distinguishing target from non-target cells, so the ones you were really looking for versus others. And so they were slightly lower, significantly lower in terms of cell abundance. Similarly, other challenges are things like these are flow cam images from the same paper where they enumerated both live cells and then the empty fecae. So we're working, have made huge strides, I'm going to say, in the last 10 to 15 years towards characterizing community composition. And actually, one of the conclusions of this paper is combining the particle counting methods with the flow imaging type methods is really the best way to go. Challenge number 2 is what type of biodiversity do you choose to characterize? Not all species are functionally, evolutionarily, or ecologically equivalent, so traditional measures of diversity may not capture relevant variation. And honestly, this really depends on the question you're asking. Right? What are you working with, and what question do you want to answer? The reason this is incredibly important is that there's no real, at least for the A, G, and C from this one study, there's really not always a clear relationship between species diversity, and here I mentioned she had the Shannon-Weaver index on the y-axis, and cell size-based functional diversity on the y. So you can't rely on the functional diversity and the species diversity being equivalent in all ecosystems. Okay, let's talk about models. Not that kind, not that kind, but this kind, right? I'm gonna get ahead of them. It's dry up here, or there's something in the air, I don't know. Right. Models allow us to predict how ecosystems will change as ocean environments change. And we already know that there's some major abiotic stressors in changing environments, things like nutrients, pH, temperature, sometimes light. And as environments change, so will the phytoplankton community. We know that. This could be in terms of species composition, cell size distribution, or physiological functions, and so will our estimates of biodiversity. So I guess when we're thinking about ecosystem models, which is one of the tools that many of us use in thinking about the ocean, what kind of diversity should we incorporate, and how diverse do we need to be? So many models of climate effects, I'm going to go big scale now for a minute, global climate and effects on ocean ecosystems, many of these use the functional diversity approach. They either use a cell size-based physiological function, or they incorporate these phytoplankton functional types that have differing functions in terms of their growth responses to something like temperature, for example, or other pH or other environmental drivers. So I'm going to describe this a little bit better than I did at the beginning. What I mean by these PFTs are conceptual groupings of phytoplankton that have ecological functionality they have in common, either in terms of the food web or biogeochemical cycles. And of course, these I mean things like nitrogen fixers like trichodesmium, calcifiers, DMS producers, their silicifiers, these things. And sometimes what you will find in the literature is that folks have used either the functional group name, so like the diatoms, or try it again, the nitrogen fixers versus the calcifiers, or cyanobacteria versus occolithophores. Okay, so sometimes the algal group jargon and the functional group jargon come together. So I threw this up here for Raleigh, but he's not here. But that's okay. Everything's fine. But a bunch of years ago, Raleigh and Ed Laws got a group of folks together to talk about this idea of pelagic functional group modeling. So note that this was published back in the dark ages, 2006. And we got together and talked about the state of the art and major challenges in current efforts to incorporate biogeochemical functional groups into models. I think I can conclude that we have successfully done that now, and it's way more routine than it was, at least at this point, in our scientific pathway. So what I'm going to do is I pulled one example from the literature to share with you. There are so many, and this one is one of my favorites. I use it in my class. So let me just go here. So this is a group from Tatiana's lab, and I don't know if the first author is actually here. No, like, okay, good, okay, thank you. I don't know if that's good or bad. Probably that's good. I'm just going to say all good things. But where they took marine phytoplankton functional types and they looked at the response to thermal change, okay, temperature variation. And I'll tell you all why I like this here in just a sec. So here are the highlights. They used diatoms, dinoflagellates, cyanobacteria, and coccolithophores. Okay, so 4 major functional groups. They asked 2 questions really. How does the growth rate of each group change with respect to temperature? And they characterized using data some of these thermal reaction norms. I tend to call them thermal response curves. It doesn't matter, it means the same thing. And then they asked the bigger question, how might these responses affect their geographical distribution under a future climate change scenario? I think this is a great example, first of all, of combining observational data with a predictive model. This was a model of sea surface temperature. I also love that they compared their results to some of another formulation. This is the FLE72 formulation, which for many, many years, modelers have been using to describe the response of phytoplankton growth to temperature. And I'm going to show you that here in just a second. I think, yeah, here it is. So back in many, many moons ago, Dick Epley published this in 1972, where he very, very early on recognized that phytoplankton differ in their growth responses to temperature, even among algal groups. So if you look, let's see, he's got 1, 2, 3, 4, 5, I thought there were 6, doesn't matter, diatoms are on the left. And these are the growth responses on the Y and temperature on the X axis. Over here on the right, he used 1, 2, 3, 4, 5 different species from different groups. So this, boy, and the names have changed. Coccolithus huxleyi is Amelia huxleyi now. Serratium is still Serratium. Gymnodidium, who knows what it is. And a couple of green algae. But using these, 7, 8, or 9 species, he came up with this one temperature response curve that, frankly, we've been using forever. Okay? So that's another reason why I love this. Compare what Epley did to what these folks did. This is growth rate versus temperature. Look at, even for coccolithophores, cyanobacteria, dinoflagellates, and diatoms, look at all the functional response curves they were able to put into the data that they used in this model. Okay, so I like that quite a bit. And from the data analysis part, they determined these different phytoplankton functional types have very different Q10s, very different thermal dependencies, very different thermal ranges. Look, I'm getting so excited, I'm breathless. Okay, so they will not respond similarly to a degree in change of temperature, all right? I'm throwing this in real fast just to show you this is another figure from their paper. Another thing I loved about this one is that if this is the growth and this is the 20% of growth rate maximum is their lower bound, they actually parameterize in the model the fact that there is an asymmetry in the growth response. In other words, this part of the increase in temperature to mu max has a slope of this, but after mu max, the decline is really steep. Okay, so they incorporated that subtlety into the model. Here on the right are the growth rate versus temperature relationships. The dashed one is the Epley curve, the blue one is diatoms, but look how different the coccolithophore, cyanobacteria, and dinoflagellate curves are. All right, here's what they did with it. They put it into a sea surface temperature model where they were able to predict global distributions of these 4 different groups under a climate change scenario. What did they find? Well, a bunch of stuff. Here are the 4 different groups. Here is, on the left, this is the proportional change in growth. So 0 would be no change versus latitude. First thing they find is, first of all, at low latitudes, the growth rate of coccolithophores were predicted to decrease substantially, while the cyanobacterial growth rates were predicted to increase. So Mike, where's Mike? This probably gets to your smaller in a warmer world talk you're going to give later on. The dinoflagellates were predicted to respond the least. And if you go back to this, you can see how flat those growth temperature response curves are. So that kind of jives with what the data shows us. All right. So just a few thoughts before I finish. Again, all of these things I've talked about so far take current data into account to predict phytoplankton functional group responses to changes in temperature, for example. You could insert your favorite variable here, right? But one of the things that they don't consider yet is, can phytoplankton either acclimate or adapt? So I mean phenotypic changes or genetic changes. Can they adapt their growth responses to predicted changes in their environment? The other thing that I've focused so far on global scale, climate time scale, okay, big changes. Not all change is on a climatic time scale or on a big global spatial scale. We know as biologists that there are lots of environmental variability that occurs on a seasonal basis, on a weekly basis. Extreme events is a good example, right? So we're going to keep these things in mind. So I'm going to finish off with just a little bit about what we've been doing in my lab, where we've been specifically thinking about this acclimation and adaptation possibility. And we focused on this group of algae called the Cryptophytes, just one other arm of the algal phylogenetic tree, special for many, many reasons that I could get into if you wish. Before I go, I want to before I keep going, I want to mention this. Notice this started in 2015. This proposal that my co-PI, Jeff Dudek, and I wrote was in direct response to something I learned here at an OCB meeting. But I think it was Heather who mentioned, or maybe Glenn, in 2013, there was a session here on ecology and evolution. I think Tatiana Reinerson was one of the chairs and possibly others. And Sinead Collins gave a talk, and I was blown away. And I went home and, 2 years later, it took a little while, but it got funded. So, I'm almost done. All right, so we've been working on the cryptophytes. The quick answer, we asked the question, how have the cryptophytes diversified into so many spectrally different habitats? You find these things wherever you go. They're everywhere. They're in puddles, they're in lakes, they're in mud puddles. If you go out to Eel Pond, I know there's some out there. We'll need to put money on that, actually, but not right now. The reason that they've actually evolved into these really different colors of light habitats is they have these things called phycobilioprotein pigments. They come in all sorts of varieties of colors. Those phycobilioproteins allow them to occupy a niche or whichever you want to call it, that many of the other phytoplankton can't occupy because they have special pigments to absorb the colors of light available. All right. So what we've been doing with these cryptophytes is thinking very much about how cryptophyte communities respond to shorter-term variations in their environment. For example, greening, brownification. Has anybody heard of brownification? Permafrost is melting, CDOM is leaching into Arctic coastal oceans, yada, yada. These are all changes in the environment that happen on timescales that are much, much faster than climate-based. But phytoplankton still have to cope. One of my students, Kristen Heidenreich, who's now at Bigelow Labs, she did some absorbance spectra and noticed this really weird response of one of these creatures under green light. And that was one of those kind of, hey, what happened there kind of things. I'm not going to tell you what happened there, but you can come to the poster. I have a poster, shameless. This is my undergrad student, Ian. Yeah, come hear about the cryptophytes more. Phytoplankton diversity has several meanings and can be quantified in many ways. I think you've figured that out already. Species or groups will respond differently to environmental stressors, and effects on diversity really are going to differ among ecosystems. Knowledge of those drivers should be very useful in predicting an ecosystem's response to things like climate change or shorter-term perturbations. But many unknowns remain. I've thrown in these acclimative or adaptive responses. Those should help phytoplankton maintain their diversity in these different ecosystems. I forgot to say that earlier. And with that, I think I am all done. Questions?
Tammi.
Hi.
So nice to see you.
Thank you. Hi there.
Great talk. Thank you.
Thank you.
So my apologies if I missed this. I went on a caffeine run for the first minute or two.
Okay.
I wonder if you could think about how temperature, in addition to its effect on the physiology and growth, It also acts as a kind of holistic simplifier for lots of different processes, one of them being resource limitation with increasing stratification. How might that affect convolve with the physiological responses to temperature?
All right, say that again, Susanna, in smaller words for my brain.
So you showed beautifully that response.
That response curve, yep.
So that, that's ecological first principles. But there's also this idea that as in certain regions of the ocean, especially things like shelf seas, where you're getting increasing stratification, temperature actually becomes a kind of holistic simplifier, if you like. It integrates a lot of different processes, and one of them is resource limitation, so decreased mixing of nutrients into the upper mix layer. And I'm just wondering how those...
the interaction with the nutrient physiology.
Exactly.
Yeah, absolutely. No, I mean, so I'm thinking out loud a little bit here because I wasn't quite sure I understood your question. Certainly, if you're stratifying and you're keeping temperature constant, is that the idea, or you're looking at changes in stratification?
All right, our first lightning talk is by Jun Yu.
Hello, everyone. My name is Jun Yu. I'm a second-year PhD student in UC Irvine, and I'm working with Professor Keith Moore. So today I'm going to introduce a new model that we are developing on CSM2 Marble, which includes 8 types of phytoplankton and 4 types of zooplankton. And we believe this is a next-generation marine biogeochemical model, which can do coupled climate rounds. So we can use this model to study questions like how ecosystems are affected by and feedback onto the climate change. So possibly it will give us a better prediction of future changes in marine biogeochemical cycles. So this is the latest published ecosystem model with 4 types of phytoplankton and 1 type of zooplankton. So we built our new ecosystem model based on this model. This model includes 8 types of phytoplankton and 4 types of zooplankton. So for phytoplankton, we have picosize, nanosize, and microsize. So for picosize, we have Prococcus, Symphycoccus, picoeucaryotes, and small diazotrophs. And for nanosized phytoplankton, we incorporate Phytocysts, Caccolithophores, and other generic nanophytoplankton. And for microsize, we have the diatoms as the classic representatives. Besides phytoplankton, we also have 4 types of zooplankton. So we have 2 types of microzooplankton. So for the smaller microzooplankton, they are the main grazers for the pico-sized phytoplankton. And the larger microzoa are the main grazers for the nano-sized phytoplankton and also the smaller microzooplankton. And we also have 2 types of larger size zooplankton, which is the mesozooplankton and macrozooplankton. So those 2 zooplankton are the main grazers for the diatoms and also the lower trophic levels. And also notably, the larger size zooplankton, such as macrozooplankton, are the direct food source for the higher trophic levels, such as fisheries. So we believe this model has the potential to link with fishery models, so we can use this model to project future changes in fishery yields. So next is some exciting part, is some preliminary results. So we believe our model can reproduce the observed phytoplankton biomass. So those 2 figures shows the model data comparison. So on the left, the figure is comparing the observed pico-sized phytoplankton compared with our modeled phytoplankton biomass. On the right is the observe the data of the larger size phytoplankton, including diatoms, Caccolithophores, Phytocysts, and diazotroph. And on the right is the modeled biomass estimation. So it's very clear that our model can generally reproduce the observed phytoplankton biomass distributions, showing the Prococcus dominant species in the lingotropic subtropical gyre, and other larger size phytoplankton like diatom and Caccolithophore and Phytocysts, they are all co-dominating in the nutrient-rich higher latitude regions. And besides climatology, I also point out that our model can capture the seasonal variations in the ecosystems. So on the first panel is showing the seasonal variations of diatom biomass percentages. So you can see there are diatom bloom in the spring season, both in the northern hemisphere and southern hemisphere. And during the summer, the diatom bloom disappear, and instead, the larger zooplankton, such as mesozoa and macrozoa, have a summer bloom, both in the northern hemisphere and southern hemisphere. So we believe our model can capture the seasonal dynamic in the ecosystem. And finally, I want to point out our model can also capture the phytoplankton group specific entropy. So as you can see, our model can capture the group specific entropy patterns, showing the highest entropy for the smaller size phytoplankton, such as Prococactus and the Synchronous and so diazotrophs, relatively lower entropy for the larger size, such as nano-sized phytoplankton and the diatom in the oligotrophic gyres. So that's all for my talk today. More interesting results are shown in my poster, so welcome to stop by and give me some suggestions. Thank you.
Our next speaker is Diana Fontaine.
Hi, everyone. I'm currently a fifth-year PhD candidate at GSO, and today I'm going to show some work from my first dissertation chapter that's currently in revision at Limnology and Oceanography. And we're examining temporal patterns of diatoms at the Narragansett Bay Plankton time series. So as you all know, or as many of you may know, diatoms are global players. They generate nearly half of marine primary production, and they're an incredibly diverse group of phytoplankton. As phytoplankton, they exhibit rapid turnover times, and so irregular sampling can miss their ecologically relevant temporal variability and community composition. So even within time series, if we do a time series approach, we can miss the incredible diversity that exists within the diatoms because they can be morphologically cryptic, and not all species are equal. Recent studies have shown that diatoms can be incredibly small, less than 20 microns in size, and are thus likely to be missed by light microscopy. So here, we leveraged the Narragansett Bay Plankton time series using a metabarcoding approach, and we had three goals: to characterize species richness and its temporal variations, to examine the seasonal and annual shifts in diatom assemblages, and to investigate the environmental correlates of diatom occurrence within the time series. So the Narragansett Bay time series has been in progress since 1959, so a very long time, and we have weekly counts of phytoplankton diversity via light microscopy. However, like I said, here we used a metabarcoding approach, and we used a subset of the time series. We used 80 surface samples from a 6-year time period from 2008 to 2014, using a metabarcoding approach shown here. I'm not going to go through it, to get at species level identification. And we found that with metabarcoding, we identified 23 diatom genera that were not observed with light microscopy during the same period of our study. And in fact, some of those diatom genera appeared incredibly frequently. So this is a plot showing the frequency of occurrence. So it's using the presence or absence data. And we calculate a frequency of occurrence shown by the equation on the bottom left. And you can see that the bars are colored by the observed presence of these diatom genera within the light microscopy data set. So if they're black, they were shown in the light microscopy data. If they're white, they were not observed in the entire time series at all. And then if they're gray, they were not observed during our current study period. And two of these diatom genera, Menidiscus and Cyclotella, appeared in over 85% of the time series samples in our 6-year period, showing that they are frequently occurring in the time series and likely play ecological roles in the system. And so now when we look at the entire diatom community, we see that the diatom communities returned a similar composition year over year. So this is a plot showing the whole time series of our study time. And on the y-axis with the solid black line is sample composition, so showing the diversity of each sample. And the dotted line shows temperature. And so you can see that sample composition and temperature are inversely related, just happens to do with our current analysis. But what's really interesting is that you see that the diatom communities are returning year after year into a very similar sine wave-like pattern of composition. And this pattern has been shown in the literature to that indicates that these communities are stable. And so this pattern we talk about in our paper, how this highlights stability within these communities. So just a quick recap, we found that there's the frequent occurrence of these newly observed diatoms They are likely, like I said, playing unappreciated and ecological biogeochemical roles in temperate estuaries, but likely beyond, this knowledge will increase with our ability to capture this diversity. And so even if these communities are changing over an annual signal, this return in composition year after year in the same season, year after year, can influence ecosystem resilience when disturbances arise. And thanks for your attention.
Susanne, if we're ready for you, if you want to unmute.
Yes, thank you. And thank you for bringing me in remotely. I really appreciate it. I'm super sorry I can't be there in person. My goal today, we've heard a lot about what is biodiversity, what are the different levels of diversity, and Emmett very nicely described there's even levels of diversity within species. And together with my colleague, Julie Roulette, we've been asking what's the role of this interspecific variability in marine microbial ecology, specifically talking about asexually reproducing organisms. And, oh, next slide, please. And without having time to go into details, there's a smattering of demonstration that marine microbes are interspecifically variable or phenotypically plastid on a range of behavioral, physiological, and genetic markers. I was at the ASLO meeting last week, and there was a fantastic session where one of the conclusions was every single cell that's pulled out of the ocean is unique. Next slide, please. So Julie and I have been asking what is the role of this intraspecific variability in competition? And we have been using game theory because it allows us to simultaneously assess individual level cell-cell competitions, which really reflects the ecology of marine microbes. A population isn't grazed upon, or a population doesn't take up nutrients, it's individual cells. Yet you can then look at processes over many, many individuals using game theory and assess the population level outcomes of individual level behaviors. On the right-hand side, so our goal was to compare what is the importance or what's the effect of interspecific variability on the competition of outcomes and ultimately on biodiversity, because we wanted to know how does it affect species coexistence. And I have two distributions here. On the left, this is a distribution that has no variance at all, and statisticians call this a degenerate distribution. On the right, with the rainbow bar, is the uniform distribution, and I've colored it that in recognition of Pride Month, and it's the maximally variable distribution, meaning the trait variation is maximal, so any trait between can be observed within a strain or a species. Next slide, please. And I encourage you, obviously I'm not explaining the details well, to check out the papers. And if you have ideas about this and want to discuss, I would love to do that. This theoretical evaluation allowed us to make key predictions that interspecific variability imparts a competitive advantage. It promotes species coexistence, even in the light of invasions, and this is based on lots of simulations. It does not lead to dominance, so variable strains are capable of coexistence. They're not necessarily dominant in the environment, and it implies an unlimited number of variable types or species or strains can coexist. And that obviously has implications for biodiversity and resilience. For example, it encourages us to quantify trait distributions rather than averages, and to incorporate intraspecific variability in analyses and models. And it very importantly contradicts the idea that there's a selection for an optimized trait value. And this approach is interesting because it promotes coexistence based on an internal factor that selection can act on, rather than external factors such as environmental heterogeneity. Next slide. And last slide, I encourage us as a community to really embrace this variability. The image shows Achilles, and we often think about variability in our data as a Achilles heel, but really it's variability in traits is an excellent strategy for a dynamic and heterogeneous and often unpredictable ocean. And this feature or this trait explains a continuous increase in biological complexity. And lastly, encouragingly, it suggests there's an adaptive reservoir for marine microbial functions that will be important in resilience, as we've heard earlier today. Thank you.
So a lot of great talks already today, and I think what I want to do with this talk is try to maybe start the debate a little bit early and have a conversation about how we can, as scientists, kind of help prioritize our studies and narrow our scope a little bit to do tractable work. Given that we have so much diversity in the oceans, how can we kind of focus? Okay, so we know that climate change, of course, is this multi-factor process that's impacting many different drivers simultaneously. So some of the examples, of course, are enhanced stratification due to warming, ocean acidification up there, and then things like mixed layer stratification, which decreases the nutrient supply. There's many, many other factors that are also influenced by climate change that can be more regional, like enhanced eutrophication or pollution runoff. And all of these are really important for us to consider in our own regions because different factors will influence our species of interest in different regions. So I really like this Boyd et al. 2018 paper because they go a lot into the multiple driver impacts. So I also really like this figure from their metal handbook that they put together to think about how it's basically impossible for us to take every single driver and every single increase or decrease in that combination and do a full factorial experiment to understand how climate change is going to impact a species of interest. And this is just like one species, right? So these would be like a bottle full of some green alga or something. So they've come up with some guidelines in this paper for reducing our effort through sort of clever collapsing of experimental design down from the full factorial, which could be represented by, the big cubes here, or let's say you've identified 3 key drivers for your ecosystem and you can't do the full factorial, but you can reduce this space and try to do some scenario testing where you're still actually getting at the mechanisms by covering a range of a particular driver of interest. And then maybe even doing the diagonal, which helps you do scenarios for multiple drivers. So I really like this kind of framework, but I want to ask the question, what about the fact that we also have this extreme diversity that we're talking about in our ecosystem? So this is just a phylogenetic tree of eukaryotes only. Of course, we also have prokaryotes to consider, but the fact that we have just so many taxa in our system, we've already discussed a lot today. So this means that sensitive taxa who are affected by some of these multiple drivers may be replaced by more resistant taxa, and so we can have this functional redundancy effect. So increasing diversity helps maintain our ecosystems in a way that makes it difficult for us to experimentally verify how things are going to change. We can also have really complex interactions between groups, which could either amplify or dampen the ecosystem impact from these multiple drivers. And so it leads to a lot of difficulty in terms of how are we going to actually figure out what climate change is going to do to our ecosystems. So I think the overall goal that I have, or that maybe we ought to have, is that we want to understand the limits of ecosystem resiliency. And maybe I should have written resistance here instead of resilience. I really appreciate it and its talk defining those terms. But basically, we have our ball and cup model where the ball represents the ecosystem state, which is relatively stable in this configuration. But what perturbation does it require to get over into a new biological system, which is a reorganized ecosystem. And in order to understand the limits of ecosystem resiliency, it's not just a single organism, it's how the organisms interact with each other that we really need to be concerned with. So I really like this figure that shows that the drop of ocean with all of these different marine organisms interacting with each other in complex ways. So we might ask ourselves the question of who are the key players in shaping ecosystems that we should spend the most time doing experiments on. So we might say the most abundant organisms, although I think that this, because of functional diversity, that this could lead us down the hole of not really understanding how ecosystems are going to change. We might also go after organisms that have the most interactions in this network of complex interactions, or organisms that maybe supply key ecosystem services, such as like fixed nitrogen. So I kind of want to approach this problem from thinking about kind of the second two, those with a lot of interactions and potentially those that supply key ecosystem services. So there's been some interesting thinking on how you get dependencies in the marine ecosystem, and this is just an example of adaptive gene loss that comes from the Morris et al. 2012 paper, looking at how cyanobacteria have lost the catalase gene. So the red dots here indicate loss of that gene from those groups. Catalase is a gene that's important for, or it's an enzyme that's important for dealing with hydrogen peroxide. And so they end up relying on heterotrophic bacteria to do this essential ecosystem function. And we can call those heterotrophic bacteria helper bacteria that serve the ecosystem. So you can see that a lot of organisms have lost this gene and that they have developed a dependency on helpers to do this ecosystem function. We had a paper in 2018, which I'll just summarize quickly, but basically we showed that just looking at one driver of climate change, which is looking at carbon dioxide concentrations in the water, we found that the interaction between Prochlorococcus and Ultramonas was altered under elevated CO2, where there was a decrease in the catalase expression in Ultramonas and an increase in, and also a decrease in the activity of that enzyme, showing that there was a greater oxidative stress and higher rates of death for Prochlorococcus. So this means that climate change not only can affect organisms directly, but it can influence organisms that are central to providing ecosystem services, which can result in sort of an enhanced sort of the amplification of climate change across the network of interactions. Okay, so this is just one simple example and just one, just two bacteria interacting with each other in a tube. But how can we potentially go about this and maybe identify more key taxa in the environment? Well, I guess I'm going to argue two different ways that I've been thinking about, and those are targeting these high-cost functional genes like catalase that can impact the ecosystem structure or biogeochemical cycles, and nitrogen fixation or the NIF genes would come to mind here also, or genes involved in iron uptake. And then also maybe if we don't consider functional genes and we back up, let's consider that trophic interactions and that web of trophic interactions, as we see here with things eating each other and exploiting each other, could also be really important for ecosystem stability. So we probably want to think about using observations to tease apart microbial interactions, and we can use microbial co-occurrence for those. Okay, so there's been lots of work on this stuff already, right? This isn't really new. I think I'm just arguing for a shift in our thinking about how to interpret. There's some cool papers that have come out about this Delmont et al. Paper looking at metagenome assembled genomes and uncovering that there's greater diversity of nitrogen fixers than we previously knew. So here is a tree showing that the diazotrophs that we have in the cyanobacteria that are responsible for nitrogen fixation are just one part of this larger tree that includes a lot of heterotrophic bacterial diazotrophs. And this is like unexpected and exciting diversity that probably leads to more stability of this service that's provided to the ecosystem than we might have previously expected. And there's some cool stuff here showing that the heterotrophic bacterial diazotrophs actually are more abundant in at least MAGs than cyanobacterial diazotrophs in a lot of the size fractions from the Terra Oceans data set. We've done a little bit of looking for the catalase gene in the Terra Oceans MAGs, and we've also found that there's a lot of diversity in groups that seem to carry this catalase gene. So you might not see this if you look at the metagenomes and just make a phylogenetic tree of catalases, because they seem to be like horizontally transferred quite a bit. So by looking at MAGs, we can tell that they are actually found across a range of groups. There's also variation in the number of MAGs that we detect in different regions. So that may suggest that there could be regional variation in how robust or resilient these groups are to climate drivers as well. Okay, so totally switching gears again, I'm going to talk about how should we identify these key taxa, thinking about observations of microbial co-occurrence instead. And again, there's been a lot of great work on this, and I don't have time to summarize it all, but some of the work, like for example, in this Neenam et al. 2018 paper, shows that by using just observations of microbes, either across a time series or across space, you can find that there are correlations between certain microbes, and they can pick apart interactions that we know from other studies. For example, the Eucinae cyanobacteria, that's a nitrogen fixer, and an endosymbiont of the Bradysphaera bigeloii. That's really exciting that we can take environmental data of time series or spatial survey and pick out real interactions from that, because there's many, many more interactions that are occurring in the oceans that we don't have experimental evidence to validate. And so being able to pick them out of these time series or spatial surveys is really powerful. There are also, this is a paper from the Terra Oceans folks, the Lima Mendez paper, showing that you can even pick out novel interactions by using co-occurrence networks, where they found that there's an ACL flatworm that was highly correlated with green microalgal eukaryote, and that turned out to be an endosymbiont for that flatworm that they could further observe by using microscopy. So this is a promising technique. There probably are lots of spurious correlations as well. I want to be clear about that it's not always going to give you real interactions, but it's a good place for us to start to be finding interactions that could be ecologically significant. I'm starting to do this type of work with my system in the Northern Gulf of Alaska. We have a 4-year time series so far, looking at samples from off of Seward, off of Kodiak, and off of the Middleton Island area. And so far, what we've been able to construct are some preliminary co-occurrence networks, looking at bacteria in blue, archaea in red, and eukaryotes in green. And the co-occurrence network structure differs by region, and we're still piecing apart what the correct chunking is of these regions to make sure that we're not including groups that are not actually, basically the narrower your region is, the more samples you're excluding, but the wider regional net you cast, you're probably losing true interactions. One thing that we are kind of just starting to look at as well is looking at these taxa that have high degree centrality, which is basically just a metric of how connected they are to other organisms into this interaction web. Those are also the taxa that tend to have relatively low read abundance. So again, I'm showing here in red archaea, in blue bacteria, and in green eukaryotes. And there's not a strong trend either way, but the ones that have the highest degree centrality are also relatively low in the reed abundance, which is kind of what you would expect for an organism if it is like a keystone member, a keystone species. All right, so I guess I want to say these are probably not the only two ways to try to solve this problem, but there are some ways that I've been thinking about. But I think that as a community, I'd love to have a discussion in our later debate about whether we can identify keystone microbes, whether we should basically focus our efforts to culture more representatives that may be keystone members of the ecosystem. That's obviously easier said than done, especially if they are dependent on other organisms. Observations, I guess one low-hanging fruit is more observations of how perturbations such as marine heat waves or storms influence the keystone microbe abundance and diversity would be a good way for us to sort of check ourselves to see if they are actually contributing to resilience. And then I'm not a modeler, but I would love it if there were more modeling experiments to understand how removing keystone microbes would impact the resilience of the ecosystem. All right, so yeah, just bringing us back to this model, the ball and cup model. Basically, how can we reduce our experimental space? So how can we figure out how to do tractable and ecologically relevant studies? And I think that some of this may lie in trying to identify keystone microbes and prioritize studying them and their impacts on the ecosystem. And also potentially maybe finding regions that may be more vulnerable to a state shift in the ecosystem, because this isn't, like Emmett said, I really loved when he said, this isn't academic. People's lives really are impacted by a state shift in an ecosystem, as we saw in the Gulf of Alaska. And so I think that we should be focusing our efforts to ID where those state shifts may occur and sort of the systemic risk. All right, with that, little time, but I would like to acknowledge my co-authors and also the Northern Gulf of Alaska Long-Term Ecological Research Program and the PIs and the captain and crew of the RV Sikuliak for making some of that work possible. And then also the acknowledgements that we work on the ancestral lands and waters of the Dene, Eyak, and Sugpiaq Alutiiq peoples, and we're striving to be in better relationship with them. Okay.
Questions?
Great talk. Love the networks. I've seen that relationship between rare species, or the keystone species often being the rare species in a community before in other networks, and I had dismissed that previously as, well, rare species are more common. So of course, if we just say there's like a random distribution of, where the keystone, how abundant keystone species are, of course they're going to look rare. Do you think the pattern that you're seeing is real beyond that? Are they like more likely to be rare than you'd expect from random chance?
Ooh. I don't know. I haven't thought about it enough, but yeah, I think that, well, we would expect keystone species to not be the most abundant thing, but there's plenty of rare things that are not going to be the keystone species. I think what I'm concerned about is a lot of false positives in this kind of analysis of things that are just co-occurring but not truly interacting. And I'm not sure how to deal with that problem, and I would love to have discussions about it. Yeah.
One more question, if anyone's interested. Hi, thank you for the talk. So you showed the tipping point curve, and now there is a lot of discussion in earth science about like rate-induced tipping points, like that not only the magnitude of the perturbation is important, also the rate. And I wonder if it's discussed in diversity studies or.
That's a really good question, and I haven't thought deeply about that, but obviously it would really matter in terms of abilities of organisms to either be brought into the system, like advected into the system that would fulfill those niches, or or be able to adapt. So it should really matter, the rate, but I haven't really represented that here at all.
Great, thank you, Gwen. Great, so first up we have Jun from Johns Hopkins University.
Hello, everyone. My name is Jun. I'm a PhD student from Johns Hopkins University, working with Ann Ehrlich. These are my collaborators. I'm going to present one chapter from my thesis, but I will also present other chapters during the poster session. Hope to see you there too. Let me do a brief overview of cryptic sulfur cycling. Cryptic sulfur cycling is a process where nitrogen and the sulfur species are interacting with each other. It's super important because it involves various sulfur-related reactions. These reactions have been incorporated into our model. Now, the interesting thing here is these two reactions also utilize nitrogen species. So this one is known as chemolethal autotrophic nitrate reduction, and that one is referred to as sulfide-driven denitrification. They play a role in transforming nitrogen species. Here, the figure on the left is showing the average oxygen distribution in June in 2017 for our study area, the Chesapeake Bay. You can clearly see that hypoxia is happening. The blue dot represents the location of the Bay Bridge Station, where we have abundant genomic data. The figure on the right is the schematic diagram of our biogeochemical model. It may look complex, however, pay attention to two things here. Firstly, we have now included cryptic sulfur cycling into our model, which was previously overlooked, but thanks to genetic evidence, we now confirmed its existence in Chesapeake Bay. Secondly, we now considered sediment processes in our model. Here, these are modeling results from our newly proposed baseline model. These results are showing time depth profiles for sulfide reduction, sulfide oxidation by nitrate, sulfide oxidation by nitrite, and oxygen levels at Bay Bridge Station in 2017. You may notice that the values for these two reactions, they are still proportionate to the concentration of sulfide present. The reason for this is that sulfide is undergoing oxidation by oxygen primarily. Although I didn't show the figure here, only a small amount is directed into these two pathways. It is likely because when oxygen is present, sulfide undergoes oxidation by oxygen primarily. However, once we run out of oxygen, we start to observe hypoxia. These processes are strongly correlated with each other. And here, I'm going to show the comparison between gene abundance versus the modeling rates of cryptic sulfur cycling. Here, the figures on first row are showing observed distribution of DSRA and DSRB with depth on y-axis and time on x-axis. The colors are indicating the sum of all oxidative and reductive types of DSRAB genes in log scales. You can notice the genes are enhanced as you go deeper into the water column. C is showing model sulfide reduction rates as a function of hydrogen sulfide concentration, colors indicating oxygen concentration. D is showing model sulfide oxidation rates. E is showing model sulfide reduction rates versus sum of all reductive types of DSRA and DSRB gene, and F is showing sulfide oxidation rates versus sum of all oxidative types of DSRA and DSRB gene. We can see sulfide reduction kind of like it doesn't seem to be all that correlated with genes. However, sulfide oxidation kind of is over sort of similar range. This will be consistent with the fact that sulfide reduction is primarily occurring in the sediments, whereas sulfide oxidation is happening in the water column. So the problem could be with our model, or the problem could be the genes. The reductive types of DSR genes could come from the bottom, or the organisms carry the gene but don't use the genes. Or it could be our model has some skill in simulating oxidation in the water column, but not a case for reduction. Our results also suggest that we do need to include the simulation of sediment processes in our model. For next step, we want to get in-situ measurements of the rates of cryptic survival cycles so we can better constrain our model. In that way, we can know more about how cryptic survival cycling could affect ecosystem resilience. And also for next step, I'm looking for a postdoc, so you can also help me by emailing me. Thank you.
Great, thank you, Ree. Next, we have Kim Peppendorf, who will be talking with us about microbial energy turnover.
Okay, so thank you so much for the conference workshop organizers and the session organizers for putting together this amazing set of talks. I'm really jazzed about all these topics and really excited to be a part of the conversation here. So what I'm going to be presenting is looking at energy and nutrient cycling under elevated temperatures. And what I'm presenting here is the work of Casey Lanford, pictured here and sitting in the audience right there. So this is work that she did with me when she was a PhD student at Rasmus, and she is continuing on doing more awesome work looking at nutrient cycling and microbial energy rates. So please go see her poster. And she's doing that as a postdoctoral fellow at Scripps. So we were really interested in evaluating how we expect ecosystems to change under elevated temperature in an observational study. And I put here, we quote, expect rates to increase, because that's such a broad statement, and there's been some great introductions to that here. So, you know, that's rates to increase is going to really depend on who you're looking at and what rates you're looking at, what metabolic processes. So we're focused on the rate of energy flux and nutrient flux, particularly phosphorus. And looking at it in just a very broad sense of a ecosystem that has a lot of different functional groups in there. And we're really focused on measuring those rates because we think that those give us really critical insights for connecting the chemistry and the biology. These chemical transformations are the outcome of metabolic processes. And because there's such tight coupling in ecosystems between the chemistry and the biology, right, it's the chicken and egg, one drives the other. So rates are a way to really quantify these connections and give us predictive tools in particular for how we expect ecosystem function to change. So when I say we measured energy rates, what we were using is a method to look at ATP phosphorylation rate using radio-labeled ATP incubations. So we were using this because ATP, adenosine triphosphate, of course, is a ubiquitous energy trafficking molecule. Every known organism uses it, maybe not that exclusively, but uses that in high abundance in their cells to traffic energy around for all of the metabolic processes. And the cool thing about ATP is that it's going to represent energy acquisition from diverse sources, light and respiration. So the idea of ATP as a measure of energy, of course, has been established for more than half a century. There's a lot of really fundamental work from Dave Carl and Bossard and Holm Hansen, London, Thor, Atkinson, 70s and 80s. So the transformational thing that we added here was using these radio-labeled incubations and coupling it with an HPLC method to make this a high-throughput method. So when this was introduced in the 1980s and 70s, and since then, it's been done with like thin layer chromatography, which is why you haven't seen a lot of rate data of ATP. So we hope that people will, do a lot of HPLC runs. So here's 10 seconds in one slide of a thesis work as developmental work. So this is what that ATP labeling method looks like. So we get nice chromatographic separation of those ATP metabolites. So we can take a bottle, add radioactive phosphorus, incubate very briefly, separate it out, and measure how quickly that ATP was turning over. So we set out then to use this to compare changes in energy rates to changes in nutrient acquisition rates in a coastal setting where we elevated the temperature. So we were working just offshore from Rasmus, which is in southern Florida. Woo! Idealistic thinking there. Southern Florida, working in Biscayne Bay. And we ran two sets of experiments, one in the winter and one in the summer. So this is our temperature. We were doing 3 to 4 day incubation. So these are our observed temperature changes. So we were using an immersion cooker on our really fancy incubation system there. So you can sous vide your steak right next to your sample bottles. And so we were able to do, what we were going for was a 4-degree increase in temperature over the ambient for sort of a worst case scenario, climate change scenario. So you can see in the winter we achieved that. And in the summertime, we'd started out with an a priori cap that we'd set for ourselves of how high that temperature we were going to allow it to go. And we were like 36 degrees Celsius. Sounds like, a crazy temperature and we don't want to bake our microbes. And then the ambient surface seawater hit 34 during our experiment. So we had a small difference between our elevated temperature and ambient there. Miami has been continuing, so that was breaking a lot of records that August 2021. Miami's been continuing to break all kinds of climate records on land and in the ocean. So we now see this summer since then, temperatures like this in the surface water all the time. So these are really relevant questions. How is the ecosystem changing? So we measured a lot of things, and in a very brief answer, looking at those ATP phosphorylation rates and phosphorus uptake rates, looking at dissolved inorganic phosphorus. We've got our conditions here, ambient, warmed in the winter and in the summer. And I'll just zoom you in to take a look at just on that first day, comparing winter to summer. We had this awesome 10-degree temperature gap. So the prediction for a 10-degree increase in temperature, the very generalized idea is that metabolic rates will double with a 10-degree increase in temperature. And again, that's really generalized, and Tammi did a great job pointing out how that's really going to vary depending on who's there and where you're looking at, and again, which metabolic rate you're looking at. So under ambient conditions, comparing our winter to summer, the change in ATP phosphorylation rates was about a 2.5-fold increase with a 10-degree increase in temperature, just under ambient conditions. For phosphorus uptake rates, it was like a 10-fold increase in phosphorus uptake rates. But of course, this is, wintertime and summertime. We had dramatically different conditions in communities. So there's all kinds of things that varied. So that's why we really wanted to look within our conditions how things change. So you can see when we looked in our different conditions for phosphorus uptake rate, we got this lovely kind of increase with temperature. And when we looked at the ATP rates, it really indicates, for one, you can see we had a lot of changes in our bottles over time. And for two, what we saw in the summer was very different than what we saw in the winter. So this kind of points to the need for a lot more measurements and seeing, how things differ when you dig into how looking at individual functional groups and in diverse ecosystems. So thank you so much.
While we get our next person, our last lightning talk speaker, I have a question online. Did you use any acclimation procedure?
So that day 0 is when we started warming them, which is why we ran a 4-day incubation, because we figured we have to give them a little time to get used to their cozy warm temperatures. We had done, this is like the not the first iteration of these temperature experiments. So we had originally done some where we tried warming things up slowly versus just going 4 degrees up. For these, on that day 1, we sampled our seawater and then dropped it into 4-degree warmer conditions and then let them go for about 4 days. So that was, and if people have insights on how we should be doing this, I would love to chat with you. Okay. Tammi says there's no right answer. Love to hear that. Yeah. So, thank you.
Thank you, Cam. Next, we have Mike Roman.
Hey, I've never given a lightning talk, but I've had 8 cups of coffee, so buckle up, all right? So this slide is to remind you of the copepods, these phytoplankton and microzooplankton, that they molt and they're eaten by fish. This is a hypothesis what I'm going to try to prove are true, but if you fall asleep or I don't get through this one slide in the next 3 minutes, just remember this is also the conclusion. Warm water will make individual copepods get smaller, right? And when you increase the temperature, you're also decreasing the oxygen, right? So it's really an interactive effect of temperature and oxygen. Also, when you warm the water, you'll also promote smaller zooplankton communities. The species will change and they get smaller species. So here's some data to prove that. These are copepods from the Baltic that were raised at different temperature water, 9, 13, and 18 degrees. Just focus on the top octagons. They're the adults. And you can see that our Characterization decreases in size at those different temperatures. And they're fed excess food, so it's not like the respiration went up and they don't have enough to grow on. So I'm doing a synthesis. It's about a 5% decrease in copepod size for every degree that you increase. Does this just happen in the lab? No. Here's Long Island Sound. Since the 40s, the temperature has gone up about 2 degrees. And on the right, the same copepod at Characterization. You can see over the year in the first 3 histograms, November's a little cooler, so they're bigger. But look at the recent dates. They're all smaller at the same time this water temperature is increased. What I haven't shown is there are a couple of big copepods, Centroboges, a Calanus gets in there sometimes, they're gone. And smaller copepods are showing up. So again, related to the temperature increase or a smaller copepod community. Why is this the case? Well, copepods molt. We don't have any experience like that, except if you're the Tin Man in the Wizard of Oz, right? So all of a sudden, every few days, they fling off their exoskeleton and they get a new one. So that temperature of development has a higher response temperature than growth. So they're molting, but they don't get a chance to bulk up like we're going to do over lunch and dinner for the next week, right? So that's one reason why they're smaller, because of those differential responses to temperature. Besides temperature, right, it's respiration. For every degree of increase, copepod respiration increases about 7%. It's a logarithmic increase. So at the same time, the temperature is going up, the respiration is going up, when the solubility is going down. 2 degrees in temperature, there's less oxygen. So this double whammy, higher temperature, lower oxygen availability. What's the poor copepod going to do, right? They get smaller. So take a look at this. This is a great experiment. In the top panel, it's 25, and the bottom is 15. So notice if you just compare the 2 panels, you'll see that, like the other graphs, the ones at this late stage at the very end, female, they're larger, right, at the colder temperature. But look what oxygen does. It has the same effect, right? 0.9 is saturated at 25, 8.4 is saturated at 15. As you lower the partial pressure of oxygen, the copepods get smaller, right? So it's the same effect as temperature. And this is happening in our environment today. So here's my one shot of copepod diversity. They all look like footballs, right? Ellipsoid. They don't have gills. There's no external organ on a copepod to get oxygen. At a microscope, you can see these little pits. That's how they get oxygen, all right? And so it's the difference in partial pressure outside the copepod, the inside, and the diffusion, right, and in the integument thickness. And so this little copepod at the top, if this works, little copepod at the, oh shit, wait a second. Okay, little copepod at the top has a surface to volume ratio of about 50. This big lunker at the bottom is 5. So think phytoplankton, small size, higher surface to volume ratio. They're favored in a low nutrient environment. The same thing, and low oxygen. In the tropics, you get small copepods, and the colder water, more oxygen, you get the bigger copepods. So we kind of figured out ways to model this, and this takes some time, but respiration rate is on the y-axis, and environmental partial pressure is on the x-axis. And see above those little points what I call P-crit? After that, respiration is not proportional to the available oxygen. In other words, oxygen isn't limiting. So if you had growth, respiration, egg production, above those little red pointer, it's not related to the oxygen concentration. When you get to P-crit, where it becomes limiting, all those functions are in direct proportional to oxygen. So the big guy, the large copepod, has, if you want to find out more how we prove that, look at that paper. So it's already happening, right? Things are warming up. Biogeographic boundaries of zooplankton and fish are changing. And in your own body of water, copepods are getting smaller and the communities are changing. And so we're trying to come up with some predictive relationships to see how that changes in the future. Bigger copepods, better food for fish. If Debbie Steinberg was here, bigger poop, bigger flux, right? All right, so these are all ramifications of getting smaller. Thanks.
So please don't forget, you have a card. If you have an idea for the debate that you'd like to present, don't be shy. You don't have to even know if you're right. Just make it an assertion and we'll have a discussion about it. What would we need to do to measure this? What would we need to know to prove this?
As we're getting settled, I assume there are some people in the audience who are taking the marine chemical speciation or MarChemSpec tutorial on Thursday or Friday. And if you would like any printed copies of any of the manuals, I would like you to come talk to me or email me so we know how many copies to print, because we don't want to kill trees if we don't have to. I'm happy to print copies as needed, but I don't want to print a bunch of copies and have people not use them. The PDFs are on the Google Drive that I shared with you. So I think we have one or two of our chairs start the lightning talks.
All right, hello everyone. Welcome back for the afternoon session. Before we gear up for our debate, we're going to do a few lightning talks and one more plenary. So first we have Jacob Cram from UMSI. Yeah.
My title's changed a couple of times. Currently it is this one. The point of this talk is that biodiversity at some depth seems to precede stability, and as you remember from the first lightning talk, it's sort of one aspect of resilience in a couple of different parameters in the time series. Microbial stability in this case. So resilience, question, does a community structure function, is the idea of whether a community structure function recovers following a disturbance. And in this case, we'll look at structure in terms of which microorganisms are present, and function being any rate. And the rates we'll look at here are primary production and microbial growth. And in this talk, I'll say that resilient communities are ones that shift less. So this is, again, a metric of stability. And at this point, we're not really controlling for changes in environmental variables for reasons. Biodiversity is, I'll talk about alpha diversity, so richness and evenness. And in this talk, I'll be using the inverse Simpson index as a proxy for diversity. My question here is, do times of high biodiversity precede times of stable community structure, primary production, and microbial growth rates? The clock is really loud. So I want to re-explore this. So I wrote this paper or published this paper in 2015. It's a seasonal, looking at seasonal community structure in the San Pedro Channel. San Pedro Channel is, we'll use the mouse instead of the pointer, here, just off the coast of California. We looked at the community structure at 5 different depths throughout the water column. And the point of the paper was to look at seasonal variability. I'd like to get, but one thing that the paper did that kind of touches on this link between biodiversity and resilience was this figure here. The idea is that we're looking at pairs of samples and saying when those samples are more similar, do they seem to relate to biodiversity? So here we have 2 different samples, or two places, and more biodiverse places are also more similar. One thing that's relevant, I think, to this section is this last bit here. So we have, here we're looking at whether, well, basically what this shows here is that at these 2 points here, less change from month to month leads high biodiversity in the second month, but only at these 2 depths. But really what I want to know, if I want to say, does biodiversity lead, cause resilience, or might it cause resilience, is to ask whether biodiversity leads stability in community structure, primary production, and microbial growth. Again, we're not adjusting for environmental stability. I will say that none of these resilience metrics relates to changes in temperature. So the short answer is that biodiversity seems to lead to stability at 150 meters only. Well, it's kind of 5 meters. So on this chart here, on our x-axis, we've got biodiversity, right? More biodiverse over here. On the y-axis, we have, in this case, resilience, stability in microbial community structure. So biodiverse communities at 150 meters tend to be more stable. We see something similar at 5 meters, although it's statistically significant but less strong. That previous figure for context. Other metrics of resilience are primary production measured by satellite and microbial growth rate measured by thymidine incorporation. Primary production, we actually see this opposite of what we'd expect pattern. So here at the chlorophyll maximum, when we see a lot of biodiversity, we actually see less stability, more variability in primary production measured at the surface. And then with microbial growth at 500 meters, when there is higher biodiversity, we see more stability in microbial growth rates. I know it's compared to that last figure, we're looking at the directions of the signs of these tend to be similar, but which things are significant is different. So in conclusion, resilience relates to diversity at some depths, right? So at 150 meters, high biodiversity precedes times of community stability, and also at 5 meters, but at other depths it doesn't, right? So this is a unique to specific depths, not a universal pattern. Biodiversity at the chlorophyll max leads to precedes variability in primary production, and biodiversity at 500 meters precedes stability in microbial growth rates. Richness, rather than inverse Simpson's index, does not appear to relate to these resilience metrics. I didn't show you that data, but I guarantee you I did the analysis. So in conclusion, temporal stability is a potential metric of resilience, and it seems to sometimes, but not always, relate to biodiversity, at least in this particular data set. Thank you.
Thank you. Next, we have Graham McGilchrist.
Hi, yeah, thanks. My name is Graham. I'm really grateful to be here. This is my first time at OCB, so grateful to the organizers and particularly for allowing me to come and present some recent work. I've been based at Princeton for the last several years, but I am about to move to St Andrews in Scotland. I have postdocs available, so if anyone's interested, let me get in touch. I am going to present some work where we have been working with one of GFDL's most recent Earth system models, a global model at half-degree ocean resolution, with the cobalt biogeochemical model, which is a 33 tracer model with a prognostic ecosystem. We've been performing what's called perfect model experiments with this to look at the limits of the predictability of aspects of the Earth system. The basic essence is that we take a 300-year control simulation, and at various times through that control simulation, we spin off initialization forecasts. So we flap the butterfly's wings by adding a little small, tiny perturbation to the initial conditions, and we allow the ensemble members, in this case 10 ensemble members per start date, to evolve and diverge over time. And then the statistics of that ensemble tells us something about the limits of predictability of the model system. So you can see this example here of this subset of the 10 here. And so we can measure predictability based on, for example, this characteristic. There's various ways that you can measure the predictability. This is something called the potential prognostic predictability, which somebody just invented to make it hard to say, which is basically a measure of the ensemble spread. You can see I've highlighted these arrows, the spread in the ensemble as a function of lead time and the sort of background spread in a control simulation, so the various possible states that the model could, that a particular variable could hold. The predictability, or the PPP, looks something like this. So this is the PPP for NPP, which is the global average. It can vary between 1 when things are perfectly predictable and 0 when things are not predictable at all. In reality, there's a statistical threshold below which you would say the system is not predictable. So this is the global average PPP for a variety of metrics: temperature, pH, oxygen, surface oxygen, and net depth integrated net primary production. That point at which the PPP drops below the statistical threshold line is what you would say is the timescale or the predictability horizon of the system. So in this case, you can see that it's somewhere between 1 and 2 years in a global average. This is notably down relative to some previous work that did this in a previous generation of GFDL's model, where they saw timelines more like 2 to 3 years for predictability of primary production and other quantities. So this is a global map of the predictability time horizon, so the time at which the PPP drops below the threshold. This is smooth to take out some of the seasonal variability, and then the PPP is smooth to take out some of the seasonality, and then the time at which it drops below the threshold is what's plotted here. You can see that in certain regions, we get a very, very long timeline of predictability, up to a decade and beyond. The simulations themselves are only a decade long. Notably, these regions, or some of the regions where there's high predictability, do coincide with regions where there's not a lot going on. There's not actually a lot of primary production. Often the regions where there's more primary production have shorter timescales of predictability, but don't let the color bar fool you. These timescales of predictability are still relatively long. These are seasonal timescales for potential predictability in the system. This is another perspective on this. I'm now looking at, these are different sectors, the Pacific, Atlantic, and Indian sectors, where I'm taking the zonal mean of the potential prognostic predictability. And on the x-axis here is the lead time here, so out from 0 months to 36 months. And you can see the drop-off. Where there's hatching here is when it drops below the threshold. So you can see the ways that at different latitudes you have different persistence of predictability through the system. I thought, given the nature of this session, I would also look a little bit more at the species composition or the species structure. There's 3 different phytoplankton groups in the model: diazotrophs, large phytoplankton, and picophytoplankton. As I was reflecting on the talks earlier, I realized that this is maybe not so much of an interesting metric. You can see that they all look fairly similar, but one of the things that earlier conversations or the earlier talks made me think is that actually perhaps the interesting question is not so much whether species diversity is predictable, but rather whether the species diversity lends itself to predictability in other metrics, such as primary production, because of the resilience in the system. So that's a question that we can address with this model set. This is also zooplankton biomass, which again shows a fairly similar structure. So that's all I got just now. My talk is actually more related to the high latitude stuff, same model experiments. I've got more detailed pictures of the high latitude stuff, but happy to show any other metrics that people are interested in, everyone's favorite region that they can look at. Thanks very much.
Great, and to wrap it up, we have the wonderful Victoria Coles.
Yeah, I gave myself a lightning talk. If you can't, do these things, then. All right, so this is my very brief take on asking the question of what kinds of models can we use to ask questions of diversity and resilience. And this was motivated actually by this recent paper by Mike Lomas and his colleagues, many OCB friends, in which they showed that at the Bermuda Atlantic Time Series station, there's been a recent trend of a decline in nitrogen, phosphorus, and net primary productivity in the upper ocean that are caused by an increase in temperature and stratification. And so they attribute, well, counterintuitively, and I think this is important for this club, carbon export didn't change in response to these trends. That's pretty surprising, and they attribute this to changes in the community structure in which there was a strong decrease in the small eukaryotes. And I thought, well, can we model this? So I put together a very simple one-dimensional model, and I increased the stratification by reducing the mixing over a 20-year time period. And as one might expect, you see a decline in the surface ammonium and a decline in the surface DIN. So this is depth on the y-axis and time, which, there, years 22 to 40 on the x-axis. All right, so here's what I wanted to do is really, my things all fell off. Oh well, that's life. What I wanted to do was compare a functional group model. Thank you so much, Tammi, for explaining functional group models to a model that more explicitly represents diversity. So in that functional group model, we have all these specific biochemical traits allocated to each organism. And in this particular case, I added 5 different sizes in each functional group so that there was some variability. And I took the exact same functions, identical ones, which would have been in that gene function library, and randomly allocated them to organisms of different sizes so that they could combine these functions and traits and have more diversity in their function. And then we build this really big organismal library, 2,000, 3,000 organisms, and we put them in the ecosystem 200 at a time. When one dies, we add a new one. So those are the 2 experiments. And we get different results, even though we're using the same functions and the same environmental responses to those functions. So in the functional group model, over this time period of stratification, we see a decline in the diversity. And in the genome model, we see an increase in diversity in response to this oligotrophication. And both models actually respond the same to a sharp perturbation. So that seasonal mixing, where you get that big spike of nutrients in the upper ocean, drops the diversity transiently, and then it recovers on a seasonal basis. So they both respond to short time scale, the same. So what drives this? Well, in genome, what we're looking at here is time on the x-axis, and the color is like a histogram of the size distribution. So the peak biomass is in the darkest colors. And you can see that over time, the genome model, the size distribution shifts from a fairly large, kind of centered at a larger size, to an even distribution of large and small plankton. And that's actually increasing the diversity. And in the functional group, it's got 5 different sizes of each functional group, but they don't change over time. So there's not a reallocation of biomass between those different functional groups. So what are the impacts on the ecosystem? So if we look at upper ocean surface nitrate, DIN, over time, what we see is that the more diverse model actually has lower total. You can't see, I hope I made the numbers too small for you to read. The upper panel has lower overall nitrate. But the change over time is smaller in response to that long-term pressure of 20 years of increasing stratification. And then when we look at the functional group model, it has a much sharper drop off, a much larger magnitude over time. So having learned, we are now calling this reduced stability, not increased resilience, and increased stability in the genome model. So we don't know which one of these is right, and we don't even know what the real world looks like in response to this. So how could we even tell if these functional group models, which are what we use for climate modeling, are actually representing the changes in diversity over time? And it may matter, because these are actually major differences in the biogeochemistry in response to these different methods of representing diversity. So I'm just leaving you with the different species in some of this over time, and you see not a reorganization in the functional group model, but a lot of reorganization in the other one. So that's the question I want to ask. How do we do this?
All right, thank you for the organizers to invite me. Today I'll tell you the example of, so we've talked this morning, we've heard about disturbance and hurricanes. Well, I have a real case hurricane example that I can tell you about, Hurricane Harvey, which was a hurricane in Corpus Christi, but in Galveston, really it was a flooding event. It was one of those, they can't decide if it's a 500-year flood or 1,000-year flood, but there was about 1 meter of water that fell down, so a lot of water. So when we think about the ocean or any body of water, this is what a happy ecosystem would look like. You have your phytoplankton that does photosynthesis, transforming CO2 into organic matter and dissolved in particulate organic matter, which feeds heterotrophic bacteria, which then feed the phytoplankton. And when bacteria die, they contribute to the microbial carbon pump, the biological pump. And another aspect that I'm really interested in, I'll tell you a little bit about today, are how viruses are implicated into this. And some of the projects in my lab were trying to understand what happens if this equilibrium is disturbed. So Hurricane Harvey was a good example of a disturbance. So it happened in August 2017, and 13.8 million acre-feet of fresh water entered Galveston Bay. So here it's before the storm. During the storm, it's cloudy, we can see it. But right after, you can see that all this water went into Galveston Bay, and there was like this plume. So Galveston Bay is very shallow, about 3 meters depth. So very shallow, so it doesn't take much to resuspend the sediment. And the plume persisted for almost a month after the storm. And the water went from the San Jacinto River all the way through Galveston Bay and then spewed out in the Gulf of Mexico. And that impacted the water quality of the bay. So the temperature dropped, the rainwater was colder than the normal, very hot bath water that is the Galveston water. And then it went back up after a few weeks. The salinity during the first sampling was almost 0 throughout the bay. There was, usually you have a gradient from fresh water to marine water. It was gone. It was just fresh water. And the dissolved oxygen came a little after. So after the sediment were resuspended, there was not much of the phytoplankton that were able to thrive. So it went, the oxygen dropped and then it came back. So what we did over the 5 weeks following the storm, we did 5 trips on one of TAMUG's vessel, the RV Trident, and we were sampling 10 stations. I'm going to talk only about 4 today because what I do is metagenomics, and I couldn't handle all these liters. I could handle 4, but not 10 times 20 liters. And so they go from the San Jacinto River all the way to the Gulf. We would always sample same transect, so always starting station 1 in the morning, station 4 in the afternoon.
And they're not perfect, but here we have Galveston and the triangle were my pre-Harvey samples. I just had my lab, and I bought the system to concentrate viruses, and I was like, scored, it works, now I can buy more. And the storm came in, so I had my pre-storm samples. They're not directly in Galveston Bay, but they're pretty close to the water quality of Station 7. So every time I'm gonna show you the data today, I'm always gonna go from September 4 all the way to September 28. And the pre-Harvey sample will be on the right or on the left, depending on what I'm trying to show you. So what we did is we collected 20 liters, and then we filtered it through 0.8 to remove the small eukaryotes, through 0.22 to keep the microbial fraction, and then we took that 20 liter and concentrated it to collect the viral fraction. And we looked at how Hurricane Harvey impacted the microbial community composition and biodiversity, how it impacted the genomic potential, and how the viral assemblages were also impacted. So how did it impact the microbial community? We used 16S ribosomal RNA gene sequencing to do that. And here we have our pre-Harvey, where we have a lot of cyanobacteria. Usually they make 25 to 30% of the community. We have SAR11. And right after the storm, well, cyanobacteria were almost gone. They just got flushed out. And it was more of like these bacteroidetes, vericomicrobia from terrestrial environments, all this flood water that went into the bay that replaced these microbes. And then we have here on September 16, there was this wastewater treatment plant that broke, and it fell there was some water, dirty water in the bay. And we have also this spike, and we're still trying to figure out what this spike is about. I don't think it's from the wastewater treatment plant, but I think microbes, there's some that throve with all the stuff that were in the Gulf, in the bay, and I still don't know what happened. But in general, the dominant members of the marine community, they were replaced by organisms from terrestrial, sedimentary, and freshwater origin. We tried to look at the origin of these microbes, and with the wastewater treatment plant, they were all broken they were all flooding because ours are open air. But we didn't find in the metagenomes many gut microbes. But it was really what was coastal before was mainly freshwater and soil microbes. And the cyanobacteria, they were almost gone after Hurricane Harvey, but within 6 weeks, they came back. So the microbes, they can adapt and they come back to what they were before. So now if we look at the genomic potential, here we had our metagenomes, and we assembled metagenome-assembled genomes, so complete species genome. And on the left here, we have them separated based on their taxonomic identification. Are they actinobacteria or cyanobacteria or beta proteobacteria? And we see that there's not much difference, even though we saw a difference at the 16S. When we look at the general species, their genomes there wasn't much change. But if we order them, cluster them by date, then we see that there is a difference. So there was really a transition of these microbes through time. So that could be one way that microbes get more resistant to perturbation, is by having a mix of everything in the bag at all times. And when we look at the genomic potential, we see here if it's red, it means that it increased. If it's blue, it decreased. So before the storm, there was a lot of cyanobacteria and photosynthesis going on. And after the storm, it dropped and then it came back. There's some that were not really much there before. They peaked right after the storm and then they left. So carbon fixation, methane metabolism, and then others like the sulfur metabolism, all these microbes from the sediment that were now in suspension, nitrogen. So there were some of these patterns of things that there was a change at the global community. Who was responsible for that? So to do that, we looked at our mags. So here, each column is one of our metagenome assembled genome, and each line is a different pathway. And when we sort them by their species, it looks as muddled as when we sort them through time. So that shows that the whole community at the ecosystem level really was resilient. And where is this resilience coming from? That can be one of the things we can discuss during the debate. So the communities were resilient. We saw changes, but these changes, like the function of the whole ecosystem didn't change. But that being said, I didn't measure any microbial activity rates, and that's probably something that we would need to measure next. One thing I want to bring your attention to, and that can be an idea for the debate, is as I was trying to understand the data, I came upon the insurance policy hypothesis, where species that might be functionally redundant in the ecosystem increase in numbers in more favorable conditions to compensate for the reduction in performance of the species, thus providing insurance for community productivity. So basically, by having microbes that can do a little bit of everything, maybe not perfectly, but if there is a disturbance, they can all still do things not perfectly, but as a whole, they can do it together. And Galveston Bay is under constant threat, not just the hurricane, but it's so shallow. We have the channel. There's a lot of raining events. So it's sort of, there's a lot of oil spills. So the community is used to these threats. So they're used to adapt to those. I have a little bit of time to talk about viruses. So just a little background of viruses. When a virus or phage, those are infecting bacteria, enter a cell, they can go through the lytic pathway, which will kill the cell, and that's usually involved in nutrient recycling. And some of them can integrate inside the genome of their host, then they become vectors of horizontal gene transfer. How did the viruses change? So here we're looking at the viral biodiversity, and on the left panel, it's from the pre-Harvey to after the first sampling, and on the left panel, it's the first sampling up to the last one. So there was a drop in diversity and in richness as well, but then it came back. So what does this mean? Well, there was so much water that didn't have as many viruses. There are viruses in clouds, just not as many as in the water column. So it's possible that there was just a drop. And this drop in viral concentration made viruses, like it was harder for them to find their host, so the infection rate may have also dropped. And it's also possible that all these changes made viral degradation faster. But when we look at viruses, so here we compare them to each other. Each dot is a different viral sequence, and we compare them to each other. So if they're in a cluster, means they share some genes. If they are separate, then they don't share any genes. And we can see that there were some groups that were present prior, that were not present before Harvey, which is in gray, like this cluster 15, only was brought in by the storm, but then by the last sampling, which is blue, was gone. Some other ones, they were very abundant before the storm, then they almost disappeared, like this 18 and 1. And then they came back in full force, and some of them just didn't change at all. These are ubiquitous. They probably infect a little bit of a wider range of a host or ubiquitous hosts. But one thing that I really wanted to mention today is that so we had the microbial fraction, and we also had the viral fraction. So we look for viral sequences within our metagenomes of the host, potential host. And we didn't see a lot of difference when we looked at prophages, but when you look at the lytic viruses, there was about the same amount before and after, but then it kept increasing and increasing and increasing. So could we use these viral contexts that we find in the viral fraction as a proxy to estimate the frequency of viral infections. That would mean that there was about 10 times more infections after 5 weeks. We didn't measure the rates, but that would mean that we would have way more particulate and dissolved organic matter from killing these microbes and after 5 weeks. So one of the things that I'm really interested in is measuring if that's true. And if it's true, how long does it take to recover? I thought that the ecosystem recovered. All the microbes look the same, but maybe the rates are different. And the Gulf Coast is hit constantly by storms. If we get hit by one storm, maybe we recover, but is there a tipping point where we no longer recover from a storm and then the carbon cycle is too accelerated to go back to what it was? So if it is something you're interested in working with me and you're a geochemist that works with carbon, find me. So, so what we saw, so the Hurricane Harvey, it really changed the water chemistry, lower salinity, lower temperature, and a lot more dissolved organic carbon, less oxygen. And this really impacted the microbial community composition, where we had this shift from more estuarine to freshwater, which came back to estuarine after about 5 weeks. The reason why we sampled 5 weeks was that salinity was back to what it was, and also we were running out of filters. So we had to decide when are we going to stop. But the microbial communities, they displayed resilience because there wasn't much change in the ecosystem function. But that's also one of the questions I had. So what makes an ecosystem more resilient than another one? Is it its biodiversity, or is it the fact that they're always used to changes and disturbances? And when we looked at the viruses, well, as we're looking that there's going to be more frequent hurricanes, there could be a long-term impact of this, especially when we look at viruses which are often forgotten or neglected in models. That could have an impact on the geochemical cycle. So just to summarize here, we had some sort of a reverse biological pump with the sediment that were back. The phytoplankton, we found less, but an increase in viral infections and also heterotrophic bacteria. I didn't talk about it, but we did measure organic carbon degradation, and it was way faster right after the storm than it was before or a few months after. And all of this work was done by two of my graduate students, Elena Woods, who is now up in Maine, and Jordan, who is graduating within the year. And it was really a group effort. During the storm, everybody was texting each other, want to go sampling? And, and it was funded by NSF, and we also got some funds with Texas Sea Grant. Thank you.
We have a question.
Well, it's great to see another, Texas talk. So question, how much does sediment play in the role, for example, buffering the microbial community system because it's so shallow?
That's a very question, and I wish we would have taken sediment sample, but it's resuspends like pretty often. As soon as it rains, you see the bay is always like resuspended. It's so shallow, and the boats are always like disturbing it. So there's always a little bit of it. So the sedimentation rate is high, but it's also always fluctuating. So that's something that would be interesting to look at.
Super interesting talk. I'd love to hear your thoughts about the relationship between viral infection driving microbial diversity. And so if there's this cycle of disturbance, diversity, how much do the viruses affect that diversity in the microbes?
That's a very good question. We did try to look for if there would be more prophages. We're like, oh, they're like exchanging gene. We didn't see that. But I think if for a while we couldn't find their host, like that's the part where the ecosystem was the most disturbed because they were not there to to kill the winter, to recycle nutrients. So you had this period of stalling in the ecosystem, and then they started again. And that's probably why they were higher, because they had to catch up on the work they didn't do for like 2 weeks or so. Yeah.
Great talk, Jessica. I'm curious, with the cyanobacteria, when they came back, was it the same cyanobacteria that came back, or was it any different?
Yeah, we didn't look at we could do some like more comparative genomics, but it was back to the same amount of Synechococcus, like same concentration. Usually it's Synechococcus we have, but that's a good point. We could do more comparative genomics between these.
Hi, Jessica. I have what I hope is an easy question. I was fascinated that you didn't find any gut bacteria there. Do you have any idea why that might be?
Yeah, because we used metagenomics, and it's not as precise, but I do know they were there because people would do qPCR specifically for E. coli. They found like a really steep increase, but there's just not as like their abundance is still too low for metagenomics to really find them.
So I actually found myself wondering if maybe, if I understood your size fractionation properly, I wondered if a lot of them were bigger than 0.8 microns and whether you actually filtered them out.
We sequenced the 0.8 and the 0.22, and we sequenced both of them.
Oh, did you? Okay, thank you.
Well, we were hoping to get a lot of phytoplankton, like large phytoplankton, but we ended up getting like all the cyanobacteria were in the 0.8 because they were attached to particles and stuff. So, yeah.
Awesome. So our debate, are you guys ready? We had great community participation. Thank you. So we're going to try to pose the questions that you guys framed as a for or against hypothesis, because so that we can have an up or a down vote on this. And we'll try this to start with. There's a lot of you, if you all came down to the front, it might take a long time. So if you think you have a take on this topic, then stand up and put your thumb up if you are pro, and your thumb down if you are con. And if you pose the question and we call your name, you can come, then we'll start with you, and we'll do a couple pros and a couple cons and see if we can come up with some fun discussion. Okay? So thank you so much to whoever, because this is the best one ever. Okay? So our question was, what does this have to do with carbon? And I mean, that's a good one, right? All right. So there was another topic similar, which really speaks up to how does biodiversity affect carbon export? Kitag. So let's frame this as biodiversity is an interesting ecosystem property, but it does not affect carbon sequestration and export in the ocean. That's the pro side. Is that the con side or con? That's the con side. And the pro side is that biodiversity does affect carbon in the ocean. And of course, some of you may feel a little wishy-washy about that. You can say that in words, but you got to pick a side to start with. Go. Who wants to stand up on this one? So if you have an opinion, if you either pose this question, and you are ready to explain your reasoning on why you think this is important or is not important for carbon, then stand up and just indicate, do you think biodiversity does affect carbon? Thumbs up. Biodiversity doesn't affect carbon export, or we haven't shown it. Thumbs down. You're going to say that when you stand up. Right? I mean, that's an excellent point, right? You can't have the discussion without framing what we're calling diversity. Yay, stand up. That's it. Stand up. You, come on.
I can't not make a comment.
Right.
Okay, here's my, if we define diversity as algal group diversity, okay, diatoms, let me just be really specific, cyanobacteria, or the other ones, or whatever they are. I would say that if it depends on, boy, I'm always going to give it depends. In other words, if it's a whole bunch of diatoms, I don't think it matters, does it? What those diatoms are, they're going to influence. And I can't believe I wrote the paper on small phytoplankton and carbon flux. But diatoms are, there's going to be more export as opposed to a whole bunch of cyanobacteria. That's I don't know if that's a pro or a con, but I'll go there. Canadians are notorious fence sitters. That's all I'll say.
Another pro, good stand up.
I'll be a pro and as sort of to add on to Tammi, but if you're harboring a diazotroph symbiont and you're a diatom and your new nitrogen, right, that's going to matter a lot in an oligotrophic ocean. So diversity matters.
I've been thinking about this biology as a mechanism to remove carbon from the surface. As a chemist, they may diverse ecosystem may retain more carbons in the surface layer, but they are very subject to the by microbial degradation. So I mean, as a very simple or very naive chemical oceanographers, I think that may not matter, but in the diverse ecosystem is definitely it's good for the health of the ecosystem. But this is unpublished work, but we did some export production analysis based upon the carbon data for the entire North Pacific. It is extremely surprising that annual variability over the last 20 years is extremely small, less than 0.1 gigaton carbon per year. This is very consistent with a very small annual variability in the net LC-CO2 fluxes. So that surprised me a lot, because this is completely unexpected, because the export production is about roughly 10 times greater than the net LC-CO2 fluxes, but very small annual variability in the export production, and also very small annual variability in the carbon in the net LC exchange. So I don't know, I do not have a clear answer, but it's my feeling.
So I'm a biological oceanographer, but I will say here that ultimately on the large scale, I think it does not matter, because carbon export is going to be limited by the amount of nutrients in the surface ocean. So it can change the variability and
Hi.
I'm getting a lot of looks here. This is my contrarian take. We'll change the variability, but the mean state will stay the same.
I think I have a slightly different take on this. I'm a geologist. I was asked why am I here in the morning, but I guess Heather, and the leaders take me on the science steering committee for reasons. I felt I'm obligated to say something. And I think this is something that we have seen numerous times in geological history. I look at organic carbon accumulation in sediments. I'm biased, but I'm going to say what really matters, what's forever, are the organics that are in the marine sediments, right? So we have seen this, and we can reconstruct in Earth's geological history what kind of phytoplankton did we have. Like, we can look at the fossils, we can also look at the molecules that it produced. It's definitely not going to be as good as quantitative as your like lab experiments or your modeling work, but we can look at them and we can say something about them. And I think it's a point that Tammi was making, and it's also in Victoria in your presentation, that the phytoplankton group are very important. That's what we saw in terms of organic carbon accumulation in Earth's history. And we actually use a specific method, that is the nitrogen isotope method, to look at phytoplankton community shifts. And we have seen that at mass extinction events, the Permian Triassic, the world's largest mass extinction events, a lot of places, the community shifted from eukaryotes to cyanobacteria. And that's associated with huge reduction in terms of organic carbon burial. And we think it contributed to the high greenhouse gas level in the atmosphere. And the warming and acidification and everything, you know, killed 97, 98 something percent of all like marine life. So absolutely, it matters.
Hello. I think it's pretty clear. I mean, I agree with other people that community structure relates to export, right? Which microbes are there, whether, you know, what big heavy ones are they not, is relevant. But then there's this other question, right? So you mentioned that, you know, are we talking about community structure when we say biodiversity? Are we talking about richness and evenness, right? Inverse Simpson index or richness scores. And my first thought is, you know, I haven't seen any, and I know a paper that says, okay, community structure relates to export, but they say that you know, Shannon index relates to biodiversity. So then I was over in my seat madly running a literature search, trying to see, is anybody saying that, you know, richness or inverse Simpson index or any of that relates to export? I haven't heard yet of any instances of people saying biodiversity, you know, precedes export or is higher in high export systems or lower in high export systems. Then there's this other question of, you know, the first one was, how does this relate to carbon? So this is the question of, does the link, and this is maybe a question as opposed to a statement, does the link between biodiversity and resilience, you know, if that link is real, does that have implications for carbon cycle, or does it not have implications for carbon cycle? My hunch is that it does, but I haven't fleshed out in my mind how that could be a link.
Yeah, the point that I wanted to make was pretty well just made now, but I just wanted to back the previous comment that no diversity would not matter. Certainly community structure does, but I would argue that the diversity does not. We've got these single-celled organisms with high rates of dispersal and very fast growth rates. And so it's not that overall, you know, is it a diverse from an evenness, richness perspective? Does that matter? No, because once that vacuum's been established, once we've left some stable state, now there's nutrients on the table to be consumed, something is going to grow up and consume that. And that might be a very low diversity in the moment, but there's that long tail of taxa waiting for their moment to come forward, and they're not going to come out in a typical diversity index, but they're there, they're waiting.
All right, we're going to move on to another of the provocative comments as we listed them. And it's, you know, obviously all of these are interrelated, and so some of this has been touched on actually in that previous discussion. There are two related comments. One was unsigned, but had the graph of productivity against biodiversity and the fact that sometimes you can see productivity decrease in a highly diverse environment. And so if we've seen this relationship before, what does that mean for the relationship between biodiversity and productivity and resilience? So I guess the two, and there was another comment, this one is actually Jeff, so you're on the hot seat again, with the thought that it's not possible to define resilience for marine microbial communities via a classical definition, because there is no quote unquote normal state to return to. And so the overall topic would then in this would be, sort of, do we know the link between biodiversity and productivity? Have we solidified that link, know its direction, or do we not know that yet? And I would say, what would we need to do to think about that? And so with that as your sort of fodder, thinking about which side you sit on with that position, which might be tough. Or, you know, again, if you're in the middle, you can also comment with your thoughts and get your opinions out there. Jeff, do you want to go first?
So pro is that you think the theory is established with observational evidence in the ocean.
Yeah, so since I wrote that comment, we've seen at least 3 examples of marine systems returning to normal states. But the system that I had in mind when I wrote the comment was, you know, something like the Southern California bite, where essentially you don't have a normal. And so how do you define resilience when there is no normal state? And this is following from that really nice example of, you know, resilience being defined as time to return to normal. Well, there is no normal state. So how do you know if your system is resilient? Resilient to what? You know, what's it returning to?
I would agree with that. And I think it's also interesting to think of from a terrestrial standpoint compared to marine, right? Like in terrestrial system, you have a landscape, you have some disturbance, and you go back and you check that landscape. We're talking about moving bodies of water, parcels of water. It's 3-dimensional. So when we talk about like resilience, something happens. Are we talking about over this flat lawn, or are we talking about in this water parcel as it moves offshore over a couple of days? I think, yeah, there's some really cool, interesting questions that we asked just from taking it out of the terrestrial sphere. And to the productivity thing, I thought in maybe Emmett's talk, there was a graph where he showed increase in estuarine phytoplankton diversity and increase in productivity. But I guess from Jacob's quick search, there aren't too many open ocean examples of that. So that's something interesting that could be explored.
This is like a tiny follow-up question. What does it mean that there is no normal? Like there's always a normal, right? You take an average, find the standard deviation, and that's what's normal. I mean,
I don't know. But you know, so we maintain a molecular time series, an amplicon-based time series at the Scripps Pier, and you've got a fairly unpredictable cycle of diatoms and dinoflagellates. You could not look at it and tell me there's a normal state with respect to who the blooming phytoplankton is. There's no standard seasonal cycle. There's no, you know, it's highly variable and difficult to predict. And so there isn't a normal state that you're going to return to. You perturb the system, you come to something new. It's probably something that's been seen before in that system, but it's not what was there before the perturbation.
So Mike Soraki sent me a paper the other day in which it noted that in eutrophic systems, we see primary productivity kind of decoupled from biomass. And so what's happening is that the primary producers are growing in excess of the ability of the grazers to take them down. So that's a bloom, right? We have blooms in the open ocean. So does that, if Emmett's hypothesis, or I won't call it a hypothesis, no, we're going to call it a hypothesis, that primary productivity and diversity are linked and primary productivity and biomass are linked, then does that fall apart in eutrophic systems? Does diversity become not a relevant indicator of ecosystem function?
So I know we talked about this a little bit when we were doing the planning for this session. I don't know, is diversity not irrelevant? In eutrophic systems, often what we find is a dominant organism, right? So when taking, you know, a harmful algal bloom or EDAB, it's something like that. So I think that the concept of diversity is still relevant in the sense that it's low diversity, right? So to me, the coastal and the open ocean are really, really different systems there's a profound thought, but that I think are acting very, very differently. I know.
Sorry, we have one online who says, Diana showed a beautiful cycle of diatom species composition over a 6-year time period. This is from Tatiana. The system has a very clear annual cycle when it comes to species presence and absence. I think by just looking at presence absence, we gain clarity on who is there when, and the abundance of the species changes from year to year, which was clouding our ability to see how patterned the system is. So maybe this is speaking to Jacob's point about richness versus diversity indices. Okay. Oh, Mike.
In this discussion, if we didn't have molecular techniques, so in other words, years ago, when you had to count phytoplankton or zooplankton, there's no question that the highest productivity export is when you have low diversity, upwelling areas, spring blooms, the Arctic and Antarctic. So this whole debate of does it relate really is kind of driven by omics. Is that worth discussing?
Hey, I don't think we have a resolution on that, but maybe I agree. I don't think that the theory is totally set, at least proven. So keep in your minds, what should we be doing to measure and prove causality, if possible, in the ocean and in experimental work for these foundational ecological theories?
While you're all thinking, I just want to encourage, obviously there's no right answer to some of these questions. So please, if this is your first time at OCB or you're, a graduate student, please don't feel shy to voice your opinion. We'll be excited to hear it.
No? Okay. No, just to come back to whether diversity matters for some ecosystem functions, I think maybe it depends. So we see the saturating curves, like with diversities in the x-axis and whatever productivity, let's say, in the y-axis, and you see that it saturates. So it seems that up to a certain level, a minimum number of species seems to be required to increase the whichever function. Not always increasing, maybe it depends. I have another question I put there for later maybe. But maybe it seems that at some point it doesn't really matter if you have more species or less. So maybe there is a threshold where it matters or not, so I don't know.
We had a lot of cards that were related to different methods that we can currently use to address the topics brought up in this session. So maybe I'll make the statement that you can be pro or against if you think that we currently have the tools to relate diversity to resilience in a more open ocean type system. Yes or no? And you can comment on what you think is needed if no.
Some of you work in this area. This is the obvious answer, but more data.
But really, there were some really great talks looking at the time scale, right, of how much, when would you expect resilience? And Stephanie Anderson has some really great work looking at the time scale of data that we have for being able to say when we observe global changes for parameters like pH and temperature, things that like we think we have a better handle on. And biology in a lot of the models is like the most squirrely thing, right? Because microbes respond rapidly and change quickly, and both evolve and adaptive responses. So really more data to know if things are changing, to be able to compare.
But Kim, how will you separate causality from correlation?
People with better statistical skills than me. It's a great question. I mean, I think really digging into the quantitative tools that we have, and there's whole fields of like causal inference that have some really great tests that usually require a whole lot more data points than we have to be able to apply them. So like the realm of social sciences has a lot of really well-defined tests for being able to determine causal inference. So that's where, again, I think more data so that then we can apply better statistical tools to be able to actually look at which things are beyond correlation, which things might be causal.
Hi, can I add something to your, that's interesting what you're saying, because it's not only statistics. Sometimes when you use statistic tools, you remove outliers that have a lot of significance in biology. So sometimes maybe you're working in ocean acidification and you don't consider the different phytoplankton blooms, and you say, okay, this data has no sense, so I'm gonna pull it out, and it has a lot of importance. So sometimes it's not only more data that we really needed, even more in the Southern Atlantic, but it's also to connect the dots and not only to fulfill everything with statistics, but to talk with your colleague that is measuring also at the same place or in the same area or have the same problems. So I think sometimes it's more simple, the answer, and not the statistical tools that, of course, are important.
Can you hear me? Yes. So disclosure, I am fully getting on to the omics train, but I feel like, so omics is this incredible tool that is allowing us to ask and answer brand new questions, as has been brought up. But we're getting so much information, and sometimes I wonder, like with metabarcoding approaches, who are we accidentally leaving out? Like there are some groups that are inherently not identified by some of these approaches, and so I guess I'm posing to those who think about this or have been thinking about it longer, how for the awesome huge data sets we're getting that we're trying to understand even just the masses of information there, what are we leaving out and how is that then biasing our understanding? And then just challenges with like using omics for abundance and copy numbers and yada, like there's just so many challenges there that still exist for interpreting these really challenging, complex data sets.
I want to also sort of follow up on that, because I think Susanne's remote talk, lightning talk that we had, pointed out one thing that is really something we should probably be doing more, which is combining omics and imaging and other, we know that there are limitations with metabarcoding, collecting things that are larger, more, or other problems. And we know that there are issues, I think, also in Tatiana's students' talk, where we saw where they've seen in the Rhode Island time series, and people have seen in the Mediterranean, that the mini discus, which is a diatom, isn't showing up in the imaging, but it is showing up in the barcoding. And if you then can dial in, it's very, very tiny and it's hard to identify, right? So I think one of the things that I think we need to be doing more is combining the different tools together in trying to answer these questions, so that, jumping on the omics bandwagon is a good way to get started, but don't forget your friends who have imaging tools like the imaging flow cytobot and the flow cam that can help you say, actually, yes, that's there, and yes, that relates to biomass.
Hi, I'm Elise Larkin. I just want to build upon this conversation in that, you know, I would say we have a lot of tools that are capable of generating a lot of data. So more data is helpful, especially in terms of regions of the ocean that are undersampled, for instance. But the flip side of that coin is in some ways we have more data than we know what to do with, right? And so it's not just about, okay, what new tools, how much more data, like how much more can we generate? It's about what questions, what very directed, specific questions can we ask about that data, and how can we integrate that data in order to achieve these sort of bigger goals, like the very first question that you posed in this debate about the relationship between biodiversity, community structure, and carbon export, right? How can we integrate this data in a very intelligent and directed and specific way, rather than just saying like, here's my omics data. Look at all these species. Wow, 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 so many species. Like, what is our research question that we're trying to address? So I would say the questions are just as important or more important than the tools themselves.
I'm going to say a few comments that we've had online. One of them is from Tatiana, and she was mentioning, related to this kind of question about methods, that we need to be able to connect diversity with rates in order to make these kind of better connections. And that reminded me a lot of what Gwen and Jessica have been saying in their plenary talks. And one more from Susanna. She was talking about when we're trying to get at this question of causality, it's often best shown with manipulative experiments, which I think is also a good point as well.
Yeah, something I wanted to point out that we saw in particularly the first ocean exports cruise is, and something I think we need to think about more as a community, is the volumes over which we sample, right? We can do metabarcoding on a really small volume, but you can have really large, relatively rare cells on a per volume basis that integrated over an aerial vertical ocean are really major players in the carbon cycle. An example of this is on exports. When we took a few liters of water, we didn't see a large diatom called Rhizoselenia. When we looked in sediment traps, they were one of the major exported diatom. And when we filtered 100 liters out of a snow catcher, we saw them, right? We don't typically filter 100 liters. So that's something to think about. And as we develop more autonomous vehicles that do filtration, they're going to be able to filter more and kind of resolve this. So it's just something I want to put out there as a lot of the omics is done on very small volumes on tight water budgets. And you're going to be just missing rare, potentially important cells.
Yes, and that statement. In addition to volumes, sequence depth matters as well, right? If you have, if you sequence 1 million things, then you're looking at there's 1 million microbes per water. Or, sorry, 1 million microbes per milliliter of water. Then you're getting things that are at least as abundant as 1 per milliliter of water, and I think it's rarer than that, which is still pretty common, right? 1 per liter is common, and you'll still be limited by your sequencing depth. Not quite sure how to work around that.
Coming back to the scale issue, we've been talking more or less entirely about microbes. And just to sort of scale this out a little bit, there are other organisms that live in the open ocean. I'm not trying to be facetious. Not very many. However, they are many, many orders of magnitude larger. And that's worth something. So the specific thing I'm thinking about is it has been suggested, and some of you probably know this better than I do, that whales, prior to industrial whaling, were abundant enough and carried enough carbon to the bottom when they died that they were a significant part of the carbon cycle. We're not going to capture any of that. To I think Bethany's comment a moment ago, you're not going to capture that in a Niskin bottle, for example, or anything else. And so, there are other processes that we might want to be thinking about that involve other kinds of organisms as well.
All right, so we've touched on this a bit already in some talks and then also some of your points through so far in the debate, and it's about tipping points. And is there a tipping point at which biodiversity can't keep up with environmental change? So, for example, ocean acidification and calcifying organisms. Is there a point at which all the calcifiers are going to die because the oceans are so acidic? And so does biodiversity really even matter in that case? A little doom and gloom, but, you know, I study pteropods, so I like them. And I just wanted to see. And then thinking about how can we measure the small signs, or can we measure the small signs of reaching these tipping points before they actually happen? So I'm thinking about Jessica's talk with Hurricane Harvey. Like, is that an example of us about to reach a tipping point and we haven't yet, but it's giving us a glimpse into what could happen? So thinking about, are there tipping points at which biodiversity basically doesn't matter?
Now it's working. Okay. I think that instead of threshold, we really need to start speaking about rates, because if we reach because phytoplankton can adapt. So if the rate of perturbation is larger than the rate of adaptation, then maybe it can have an effect. But if we are just reaching a threshold, I don't know, I think it's meaningful to speak about threshold without talking about rates.
Hi, I'm a chemical sonographer, and I can't speak for the zooplankton and phytoplankton, but checking the CO2 record in the atmosphere for the past less than 1 minute, somehow there's kind of a tipping point for at least the air CO2. Somehow this air CO2 drop and then come back. So I think there's some big picture problem regulating the entire system, and that can be called a tipping point maybe.
Hi, it's a geologist again. Well, since CO2 is mentioned, I just want to say that if you look at Earth's history, right, if you look at the dinosaur ages, CO2 in the atmosphere are thousands of ppm, maybe more than 2,000 of ppm, and today is 420. So, and during the Cretaceous, it's actually the period known for calcareous plankton. Right? That's chalk. That's where the name is coming from. And that's where you see the picture, like off the coast of southern England, there are hundreds of meters of chalk deposited during the Cretaceous. So there are a lot of differences. You know, you can think about alkalinity, it's a high alkalinity world in the ocean. But I think my point is that it's not that straightforward. It's not like a single tipping point that's going to be crossed, then all of the phytoplankton are going to be gone. But what I think what's more important is the rate, because the acidification rate, the CO2 increasing rate, has not been seen in Earth's history for at least millions of years, tens of millions of years. So the ecosystem is not given enough time to respond to that. So, well, what I just recall, you know, during COVID, people say flatten the curve. And I think maybe that's the same term that should be applied here. I mean, it's going to change. I mean, look at how much CO2 we're emitting. But if we can delay that process somehow, you know, give them an opportunity, give them a time to respond to that, to, you know, adapt, adjust. And I think that's really the thing that we can do.
So is biodiversity, then, to come back to our starting point, a harbinger of tipping points or of change in the system or pressure on the system that might lead to kind of irreversible changes? Can we harness these ideas of biodiversity to understand as we approach places where the ocean is entering a regime shift?
thought following up on the rates of change and how things can respond. And I think it's useful to think about some of the different types of diversity that people have been mentioning. And, you know, there are so many metabolic capabilities that microbes have that they can turn on very adaptively and rapidly as conditions change. So that's, but then at some point, you know, when do conditions exceed the genomic metabolic capabilities that they have, at which point you're shifting into, you know, the rise of other organisms that are in the area, and then at another point, maybe shifting beyond kind of what is in the broader three-dimensional area. So those are all different kind of perspectives of diversity that also interact at different rates with environmental change.
And I'll just add to what Kim's talking about, and kind of going back to what Emmett was talking about as well, with we're thinking like microbes, right? So microbes move quickly, they're suspended in a 3D media, they can occupy lots of different niches and come in. And so diversity defined as like this set of taxa might be different. Diversity as this function might be the same, or it might be different or not as effective or not as efficient, I think is like a good way of thinking about it. But then when we think about like organisms that have lifespans of 20 years, they can't, right? You know, so it's a very, very different kind of question that we're asking. And then Victoria had asked something about, is high diversity a tipping point, or is it a metric of this? And I've seen some papers from Terra Oceans where they're trying to identify sentinel species, specifically when you're looking at like polar regions. And so I don't know if that would be a viable way of doing that, but kind of saying, well, this is an organism that should be within this regime. Oh, we're seeing it in the Arctic now. That's weird. Is that, you know, some sort of metric like that? But I think it's always hard to put like an index on, like an ocean health index. Like that's a very difficult thing to parameterize.
Good point, Bethany. It makes me think about Gwen with the keystone microbes. And I don't know, I mean, is having a keystone species a useful way to look at biodiversity? To me, that seems counterintuitive in some ways. But I think maybe for management purposes, or, you know, especially if it's a very economically viable organism, then yes, it probably should be a focus of maintaining. And
okay. I'm not quite sure whether we can find a tipping point using this biodiversity, because it's so complicated. And then I don't know how to determine the either decrease or increase in the biodiversity, even in any one single system. But please be patient, because I'm a chemist, so I try to make things very simple. When I heard very interesting talk in the December last year AGU meeting, and he's a land ecologist. He's a Amazon forest. He clearly claimed that the 2 degrees Celsius increase turned the entire Amazon forest to savanna because of the acceleration of trans-evaporations. So it changes the water cycle. So it becomes very clear to me that land ecosystem, you can set maybe a tipping point, which is the 2 degrees Celsius. I'm not 100% sure, or I'm not sure how we can do in our ocean system. So, sorry, this is the very negative point. But
I think we have some recent examples, or we could point to how at least the Alaskan Arctic seems to be going through really rapid change in the past, even just 5 years. And, you know, maybe we can go back to some data prior to that time where we probably had pretty good data, you know, and try to take a look at if there are any indicators that preceded that sort of borealization of species moving up into the Arctic. And it might be a good way to test some of our ideas, because it could not be predictable. And I worry about that too. Like the talk that was given with the modeling, how it's not predictable greater than 2 years out. That stresses me out. I'd like to talk about that.
Just to make the comment that just because something's not predictable 2 years out doesn't mean that it's not predictable 20 years out or 30 years out. It's this distinction between interannual chaotic variability versus forced change. So you can still you might still be able to make statements on long-term change, forced change, even if you don't know, we know that it's going to be warmer in next June than it is in December because we have an understanding of the system. So yeah, interannual variability, not the same as, so don't be too upset.
Yeah, hi. I also just want to add a small comment. And when we talk about like tipping points, we shouldn't, and especially carbon cycle and carbon export, we shouldn't leave out other parameters such as the calcite saturation state. So yeah, just to keep this in mind that this also affects especially calcifying organisms.
So I'm wondering if another way to think about this diversity and tipping points is to look at other sources of change. So instead of changes to just the abiotic environment, what about from biological invasions? So we know that in certain systems like Great Lakes, Black Sea, et cetera, the largest changes have happened as a result of biological invasions. So I'm wondering if folks who have studied this or know of the literature on this, does diversity decrease the likelihood of a system change due to invasion?
Hello. We do need more data, I agree. Data can be overwhelming very often. We end up, as Emily says, with an overwhelming amount of a gigantic table with a huge list of ASVs that we don't know what to do with it. But when we think about what Emmett just mentioned, that we need to look at the entire trophic structure of an ecosystem to understand how it's changing over time, we do need a lot of data. You go to the Ocean Biodiversity Information System, you see enormous gaps of data over most of the pelagic ecosystems in the ocean. And so how do we get to a point where we can build baselines of biodiversity at different trophic levels to really start asking the question of how changes in biodiversity of the basic trophic levels with secondary producers, top predators, etc., is affecting the carbon cycling? I think we are far away from that.
I'll just ask a provocative question for fun. I think this last point that was made is if we're far away from even just getting a baseline of what a system looks like, are we going to be able to observe a baseline of the system before it changes in the context of the rapid change that's happening? And if not, then should we be giving up on trying to get a baseline measure? Should we be actually thinking about entirely different ways of approaching the science in this field if we can't hope to actually measure it before it changes?
As a fellow geologist in the group here, I feel like if we can establish some confidence in being able to use these tools in the sedimentary record, that could potentially open up the ability to go back pre-anthropogenic perturbation to get at a better baseline, but I still am concerned that we're missing important pieces with the approach that we're using.
I'm gathering my thoughts because I keep wanting to respond to all of these. But coming back to Enrique's point, and we've talked a lot about this sort of overwhelming quantity of data, which I think we have to consider a good thing. Keep in mind that pretty soon ChatGPT will have a higher H-index than any of us do. So in other words, at some point, we should be able to use artificial intelligence to really make more sense out of this large quantities of data that we have than we're doing right now. That's not going to be the answer to everything, but a related point comes back to the question earlier about, do we have the tools we need to answer this question? I think we absolutely have the tools we need. We just don't have enough money. Maybe that's not the right way to put it, but if we could do the Tara ocean expedition every year throughout the ocean, we would easily be able to answer these questions with ChatGPT or some other version of AI that is able to handle that kind of data.
So just to respond to that thought, those of you that know me know that I evangelize a lot about collaborations between domain specialists like ourselves, so all the different flavors of oceanographers, and actual data scientists. They actually have tools to do the things that we're talking about, and it's done in other disciplines, particularly climate science. And this idea, there's the very well-cited paper by Stephanie Henson about how you needed the 30 years of the chlorophyll record to discern any anthropogenically driven climate change signal. The answer to that is actually it depends. It depends on what model you're using. And if you use different ways to model the system and different analytical, statistical, and machine learning approaches, then the time scale needed to discern a real change becomes different. So I think I'm agreeing wholeheartedly with you. We as a community of oceanographers need to be embarking on collaborations, not just the use of machine learning, but the collaborations with data scientists, because we know the ecology, the carbon science, the geology. They don't, and they can often, if you just give a data set to a data scientist without any context, it's kind of nonsense. So I would appeal to the community as a whole to begin to write a data scientist into your grants and make it a 50-50 collaboration, because right now we are not leveraging the full power of AI and ML in these types of Earth system questions.
Just to answer the question about AI and the comment about AI, I'm really concerned about it, because ultimately it will work based on our paradigms, and you cannot test a paradigm just from inside itself. So it's subjective by itself, the way that AI will answer the questions that summarize the question. That's the point. If you do it for exploration, it is. But if you try to have it to answer, to verify a paradigm, you cannot use it that way. So exploration, I completely agree with you, but you still need the science. Yeah.
So we rarely talk about management, really, I guess, at OCB, except in the context of ocean CDR. But we had a couple of questions that I want to make sure we get a chance to discuss everybody's thoughts that related to explicitly managing and its impact on biodiversity. So Li Zhang, if you're here, raise your hand. Thank you. Great point, making the point that sometimes certain species are targeted for eradication in order to increase biodiversity. And so what are we doing there in terms of allowing the system to adapt to a new reality? And then we also had the issue of fisheries. So when we manage for fisheries, in essence, we are sort of shifting our biodiversity and our management of that biodiversity to favor extraction of commercially viable species. So I think I had originally a pro and con statement in my head about this, and I might be gone now, but I think Jamie knows it.
That's not exactly why I raised my hand, but it bridges the two things that we were just talking about. When we talk about tipping points, usually why I think they're important is because it's like really we're thinking about something so nonlinear that it would shock the system. Like, will the levee breach? Will the glacier become destabilized? But in this conversation about tipping points, we're just like, I don't even know what we mean. What does that mean, a biological tipping point? Like when there were 300 million beavers in the U.S., it's been a tipping point for the beavers. If the coral goes extinct, it's a tipping point for the corals. But where is it relevant to, like, what is the relevance? Where's the root of the problem of the questions we're asking? And I think that gets to this new question that you're asking is there something management relevant or societally or culturally relevant about the tipping point, about biodiversity, that's very, I don't know, that evokes some people outside this room even. And so I was going to pose it more as a question, flipping the script, right? More of a question than a comment. So.
I think we could point to things like the closure of the snow crab fishery in the Bering Sea, which happened recently and is unprecedented. So things like that, where it seems that the system has tipped into a new normal, and they're not previously viable commercial fisheries are not viable anymore. So that's one thing we could point to.
I always think about the Arabian Sea, right? And the shift from a diatom-dominated environment to Noctiluca. We've heard people talk about that here at OCB before, Joachim goes. So that's an interest, that we do see open ocean, large-scale regime shifts, and I think probably the Black Sea, and jellyfish is perhaps another environment. Everybody tired?
I think we're actually approaching the end of our debate time.
We are, but is there anyone who has like a burning idea about how we really need to reformulate the way we are measuring and monitoring our ocean to understand these ecological principles and their interaction with the biogeochemistry? Yeah.
I mean, I still feel like we're stuck on this experimental throughput limitation, and it gets back to an earlier statement. I think it was either proposed by Kim or to Kim about mechanistic. I'm losing my train of thought. Anyway, to do things to move along beyond correlation into that causation space, to understand the mechanisms, we need to be able to carry out the experiments. And even though we can do fancy hypercubes and we can do really tricky experimental designs to minimize the parameter space, we're still stuck with this impossibly large parameter space just for single taxa, let alone we're still trying to figure out how to define the taxas at metabolic diversity and functional diversity and all these other things. And we've got to explore all of that in the context of that parameter space and the interactions. And so I'm still stuck on how we're getting so good at the observing and so good at the in silico modeling, but we're lagging in our experimental techniques. And I don't know what the solution to that is.
I would also, I'm hoping that the session that's at the end of this meeting will hopefully help us with this, as we're going to talk in the last session of OCB is on sustained observing systems, and that definitely is going to help us with information on baselines. But another thing that really stood out to me in Jessica's talk at the at the end of this session was how the ability to respond to actually seeing a perturbation and not only have the baseline beforehand, thankfully, of her samples right before the hurricane came through, but to be able to respond to that and how great it was that Tammi, all the people at Galveston managed to get out there and sample, and we're, crazy oceanographers, like in the middle of our hurricane, going, you want to go out and sample? I mean, any of us who have friends outside of oceanography, when we start saying these things, our friends are like, wait, what? But, take advantage of those. And hopefully the agencies will talk to this, but, certain agencies like NSF have rapids and things like that to help us be ready to go out and respond and measure those systems in response to perturbations, in response to a singular event. And unfortunately, we seem to be having a lot of singular events that are happening with wildfires and volcanoes and hurricanes coming through, but a combination of sustained observations, knowing the tools and getting together the right team of people to get all that data, but then collaborating with data scientists to make sure that we don't just collect the data, but we synthesize it. And so there's a lot of challenges, and it's going to take a lot of people, but I think that if the right people and OCB is great at combining people across the oceanography disciplines, but, and with our broader online presence, hopefully we're pulling in people from a big range of fields that can hopefully start to answer those questions and get the teams together to respond.
So last thing, if your thinking changed about the role, it doesn't matter for or against, if your thinking changed about the role of biodiversity, resilience, and carbon and biogeochemistry in the ocean, if your thinking evolved, stand up. Yay! Okay, we did well.